Okay, excellent. All right, well, let's kick this off.
All right, are you me to go, Lita, or do you want to start with?
I want you to start with the roll call. So introduce yourself as the chair, and everybody can say here as Ginny reads off your name.
Well, good morning, everyone. Thanks for the patience already. 7 minutes into this socially distanced P&T committee meeting. I'm Virginia Bucola, or Ginny. I'm going to read off your names. My apologies if I mispronounce anyone's names. After I say your name, please call here. Starting with the P&T committee members, Alex Park.
Here.
Susan Flatabow.
Here.
Diane Schmokey.
Good morning, I'm here.
Jordan Thorhoff.
Here.
Nancy Lee.
Here.
Leah Marcotte. Here. Catherine Brown.
Here.
And Connie Huynh.
Here.
Moving to the Healthcare Authority members, Lita Avascas.
Here.
Donna Sullivan.
Here.
Ryan Pistoresi.
Here.
Chris Chen.
I don't think Dr. Chen's going to be joining us
Thank
quite
you.
yet.
Luke Dearden.
Here.
Amy Irwin.
Here.
Ryan Takatomo. We'll pass. I'll move to Marissa Tabile.
Here.
L&I members, Jamie Mai.
Here.
Here.
Magellan Medicaid Administration member is Umang Patel.
Here.
Our DERP presenters are Leila Kawadi
Here.
and Gerald Gardelheiner, pardon my pronunciation. He might not be on yet.
That's right. Yeah.
Our managed care organization representatives, Jennifer Wang from Molina.
So I will have to unmute them. Let me
Okay.
see.
So it's Jennifer, Petra, and Catherine.
Okay, I see Petra. Hi, Petra, I've unmuted you.
I'm here.
Okay, and I don't see what was, oh, Jennifer Wang, I see Jennifer, but she is not on the phone, I see.
Okay. And then Catherine Vu of Community Health Plan.
Okay, I don't see. Yeah.
I want to give a special welcome to Leah Marcotte. Leah, Dr. Marcotte, I hope I'm saying your last name correctly. She's our brand new member of our P&T committee, and this is her first meeting. So we wish it wasn't socially distanced and we could welcome you. If not to put you on the spot, but if you could share a little bit about yourself with the committee, we'd like to just say, I'll say a warm hello.
Yeah, of course. Thank you so much. I'm really happy to be here. I am a primary care physician at University of Washington. I'm also the Associate Medical Director for Population Health at UW, and very excited to be on the committee. I live in South Seattle with my partner and 2 dogs.
Welcome, and feel free to ask questions as needed. Of course, we're all here, and Lita is amazing at supporting us all.
Thank you.
So I'll move to Lita for you to go over meeting logistics.
Let me unmute. Thank you. Okay, so the committee and presenters have all been added as organizers. You can mute and unmute yourselves. Please mute yourself when you're not speaking to limit the background noise. The presenters will share their webcams while speaking, and then the P&T committee will share their webcams during discussion and motions. We did realize this morning that only 6 webcams are being allowed to be shared at a time. So I realize not everyone's going to be able to share their webcam. And you can turn off your webcam when you're not presenting. For stakeholder participation, a number of stakeholders did sign up before the meeting. So during the stakeholder input section on the agenda, the chair will call the list of names that pre-registered. Each person will get 3 minutes to speak. I will unmute you. After the list of names has been called, then the chair will ask if anybody else would like to speak. You need to use the raise hand function, which over under the attendee list, you'll see a little hand. So just click on that and I'll see that you want to speak. I'll call your name and unmute you. You could also ask questions in the questions tab, and I will address those during the stakeholder input portion. Let's see. And the meeting is being recorded, so please state your name every time that you speak. And now Donna Sullivan has an announcement.
And hi, this is Donna. So I just wanted to give a slight update to the agenda. We will not be reviewing the Oxbryta clinical policy today, so we will postpone that to a separate meeting.
Okay, so I think we can have Layla begin.
All right, thank you everybody. Happy to be here today to present results from updated systematic reviews on targeted immune modulators. I'm going to be presenting 2 of the 3 updated findings, and my colleague Gerald Gartlaner will present the 3rd one. So you're going to hear from me, then you'll hear from Gerald, and then I'll be back. These are findings from an updated systematic review that was previously one large review. It was split into 3 smaller reviews focused on specific clinical conditions. This first one is related to Crohn's disease, the use of the TIM agents in Crohn's disease and ulcerative colitis. I'm going to spend a bit more time taking you through the methods on this, and then we are going to kind of skip through methods on the subsequent 2 presentations because the methods were largely the same, just for the sake of time. These were all originally much longer presentations, and we're going to try to condense them for your meeting here today. So with that, if you'll go to the next slide, is just a brief overview of the presentation. And then we'll go right into the background. So if you'll skip ahead to slide 3. So as you may know, Crohn's disease and ulcerative colitis are chronic inflammatory bowel conditions, whereas Crohn's disease involves the full thickness of the bowel wall, anywhere from the mouth to anus. Ulcerative colitis typically only involves the mucosal surface and is limited to the colon and rectum. Targeted immune modulators, or TIMs for short, are a group of medications that selectively block the mechanisms involved in the immune response. And the first TIM for Crohn's disease was infliximab, which was approved by the FDA in 1998. And since then, many additional agents, including biosimilars, have been approved for both conditions. Next slide. The TIM agents that are either FDA approved for Crohn's disease or ulcerative colitis or both, or that are in the pipeline for approval, fall into 6 categories in terms of their mechanisms of action as depicted on this slide. So some agents are approved for both conditions. Those are the ones that are depicted in the light bluish grayish color. Others are only approved for one or the other condition. Those are depicted in either light green or in yellow on the slide. And you'll also notice there are 4 pipeline agents which are not yet approved for either Crohn's or ulcerative colitis. Upadacitinib is already approved for rheumatoid arthritis, and risankizumab is already approved for psoriasis. while the other two agents, pefacitonin and PF04236921, are not yet approved for any indications. Next slide. So these are what we call the PICOS, or the study selection criteria that we used for the update. The populations for this particular presentation were adults with Crohn's or ulcerative colitis. Gerald will be presenting on populations that include persons with rheumatoid arthritis and ankylosing spondylitis. And then when I come back, I'll be presenting on the populations of persons with psoriasis and psoriatic arthritis. The TIM agents that we considered for across all three were TIMs that are FDA approved or that are in the pipeline for approval. And the specific drugs for ulcerative colitis and Crohn's were the ones I just described on the previous slide. For comparators, we selected studies that compared one FDA-approved TIM agent to another agent in a head-to-head comparison. For the pipeline agents, we also considered placebo or standard of care comparisons. And for outcomes across all three reviews, we selected studies that reported on measures of disease remission, clinical improvement, quality of life, adverse events, serious adverse events, and other health outcomes. And finally, for study designs, we were mainly focused on randomized controlled trials of at least 12 weeks or longer duration. But for harms, we also selected cohort studies of at least 12 weeks duration that had 1,000 or more participants. Next slide. This slide describes the three key questions for the update. The first question was about the comparative effectiveness of the TIM agents. The second was about the comparative harms of the TIM agents. And the third key question was about variation by subgroups. The full reports also included a question around ongoing studies, and I refer you to the full reports for information about that. Next slide, and you can skip ahead to slide 8 if you would. This is a brief summary of our methods. Again, that is the same across all three reviews that you're going to hear about today. We searched Ovid, Medline, Embase, and the Cochrane Library, picking up where the prior review had left off, which was January 2017 through last fall, with continued active surveillance of the literature through about the end of the year. We used OpenEpi for calculating absolute risk differences and incident rate ratios, risk ratios, and confidence intervals for any newly identified studies where it was not that information was not provided by study authors. And then we used the grade approach for assessing the overall quality of evidence for each comparison for up to 6 outcomes. And lastly, as I mentioned, we did look at ongoing studies, and that's available in the full report. Next slide. As a reminder, when the reviews for DERP, we do include an assessment of the methodological quality of each included study using a standardized assessment that results in a rating of good, fair, or poor methodological quality for each study. And the definitions of those are on this slide for your reference. And on the next slide is just a reminder of the grade approach for evaluating the body of evidence for each comparison. So in this review, we looked at up to 6 outcomes, including disease remission, clinical improvement, adverse events, serious adverse events, and withdrawals due to adverse events. And the evidence is judged with respect to consistency, precision, study limitations, which is essentially methodological quality, and directness. Bodies of evidence for a comparison and an outcome are then rated as high, moderate, low, or very low quality. And bodies of RCT evidence start by default at a high rating, and then we downgrade them 1 or 2 levels for any concerns in any of those domains I had just mentioned. Bodies of observational evidence, such as cohort studies, start at a low rating, and then they can be downgraded for those same domains, but they can also be upgraded when other features are present. And this slide just shows how each grade rating can be interpreted with respect to confidence in the findings. Next slide. So now we'll move into the specific findings for ulcerative colitis and Crohn's disease. This is just an overview of our literature flow diagram. Basically, the important part is at the very bottom. We ended up identifying 3 new studies and carried forward 6 studies from the previous review for a cumulative total of 9 unique studies that were in this update, and 2 were conducted among participants with ulcerative colitis, 4 were conducted among participants with Crohn's disease, and 3 were conducted in mixed populations that included participants with both conditions. On the next slide is an overview of the 9 included studies. As I mentioned, 3 are new to the update, 6 were in the previous report. 4 were randomized trials, while 5 were cohort studies. And you can see some of the other study characteristics summarized on this graphic. Of note, 8 of the 9 studies were head-to-head comparisons of TIM agents, and 1 was a pipeline study comparing a TIM agent to placebo. Next slide. Before we get into the specific findings, I just wanted to provide you with a key to some of the abbreviations that will be encountered both in this presentation and in the other 2 you'll hear. I won't read them all off the slide, but they're here for your reference in case you want to refer back to them. So first we'll discuss Crohn's disease. If you'll go to the next slide. One more, please. Thank you. So for key question 1, which is about the comparative effectiveness of TIMs, for Crohn's disease, we identified 1 poor methodological quality study and 1 fair methodological quality study. The poor study, quality study was an open label, randomized persons who were post-ileocolonic resection and at high risk for recurrence. They were randomized to either adalimumab or infliximab. And in this particular study, there was no difference in clinical, endoscopic, or histologic recurrence after 12 months, and we graded that evidence as very low. The fair quality study was an open label switch trial that randomized persons taking infliximab maintenance therapy to either continue the current regimen or to switch to adalimumab with follow-up over 54 weeks. The median scores on the inflammatory bowel disease questionnaire, which is a quality of life instrument specific to inflammatory bowel disease, were not different between the 2 groups throughout the study, and we also graded the evidence as very low. Next slide. For key question 2, comparative harms in Crohn's disease, in addition to the 1 fair methodological quality I had already mentioned, we also identified 3 cohort studies. Each used slightly different comparisons. First, 1 compared adalimumab to infliximab, which that's from the fair methodological quality RCT, and that's listed on the lower panel of this slide. For that study, for that trial, for adverse events, the relative risk was 1.1 with confidence intervals that included the null effect, so essentially no difference between groups in terms of adverse events, and we graded that evidence as very low. For serious adverse events, the relative risk was 10, but as you can see, the confidence intervals, it was very wide, ranging from 0.57 to 174. So really, you can't make much of that relationship. It's just too imprecise. We also graded that evidence as very low. For withdrawals to adverse events, the relative risk was 6.2, but again, the confidence intervals are way too imprecise to determine the relationship, and so we also graded that evidence as very low. And lastly, for infections, which was an outcome reported in 1 cohort study, the adjusted hazard ratio was 1.6 with a confidence interval that spanned the null effect, but was also somewhat imprecise. So we also graded that outcome as very low. Moving on to the other next slide comparisons for from cohort studies, there was 1 study that compared adalimumab with certolizumab and infliximab. So there were 3 study arms there, and there were no statistically significant differences in serious infections between any of the 3 pairwise comparisons, as you can see in the top panel of this slide, and all of the estimates included the null effect. In the bottom panel of the slide is a comparison of adalimumab with infliximab and etanercept. This particular outcome was reported in 2 cohort studies that were conducted in mixed populations, meaning the populations included participants with both Crohn's disease, but also included participants with other autoimmune diseases for which biologics are indicated, for example, rheumatoid arthritis. The outcome that they reported was tuberculosis incidence, and there was no difference in the incidence of tuberculosis between adalimumab and infliximab, which is the most relevant comparison here since those were the drugs that are approved for Crohn's disease and ulcerative colitis. There was a statistically significant higher incidence of tuberculosis for adalimumab and infliximab when compared to etanercept, but I know etanercept is not FDA approved for ulcerative colitis or Crohn's, so it's a less relevant comparison. Next slide. For key question 3, which was around variation by subgroup, we did not identify any studies reporting any subgroup data, so that didn't have any evidence there. So we're going to skip pipeline TIMS just for the sake of time. They all are in the full report, and we are going to move on to the findings in ulcerative colitis. So this would be slide 22. Thank you. So for comparative effectiveness in ulcerative colitis, we did identify one new fair methodological quality RCT that compared vedolizumab with adalimumab. This study reported a higher incidence of clinical disease remission and endoscopic remission at 52 weeks for vedolizumab compared to adalimumab, and we graded this evidence as moderate quality. Steroid-free remission was numerically lower among participants with vedolizumab. As you can see here, it was 13% versus 22% among adalimumab participants, but this difference was not statistically significant. But it did come close to excluding the null effect. The upper confidence interval was 0.4%. We graded this particular outcome as low quality. And then lastly, participants that were randomized to vedolizumab experienced significantly larger improvements on 2 different quality of life measures compared to adalimumab. And so we graded that evidence as moderate quality. So bottom line on this comparison is that vedolizumab seems to be more effective than adalimumab on most measures. In terms of comparative harms, we identified 3 comparisons. The trial that I just mentioned, which compared vedolizumab to adalimumab, and then there were 3 cohort studies, and I'll discuss each of those in turn. In the trial, which is at the data is shown at the bottom panel of the slide, which compared vedolizumab to adalimumab, there was no difference between groups for adverse events and serious adverse events or withdrawals due to adverse events. And we assigned a grade of moderate to overall AEs and then low to the other 2 outcomes, primarily because those 2 outcomes had much rarer events, and then thus the estimates were more imprecise. In the next comparison, which is infliximab versus adalimumab, we identified 2 cohort studies. One reported on the risk of serious infection, and the other reported just on the risk of infection. Both reported a lower incidence of infection with infliximab. The hazard ratios were 0.62 and 0.68 respectively. But as you can see, both of these results were imprecise and not statistically significant, so we graded this evidence as very low quality. The last comparison was between adalimumab and infliximab. Sorry, you can go back to the previous slide.
Sorry about
yep,
that.
Thank you. Yep, the last comparison was between adalimumab on the lower panel of the slide, infliximab and etanercept, and these are the same 2 studies I talked about in the Crohn's disease section. They reported on tuberculosis incidence, and as I mentioned, there was no significant difference in TB incidence between adalimumab and infliximab, which again are the 2 drugs approved for these indications. Next slide. There were no subgroup findings for in studies among persons with ulcerative colitis to report. And we identified several relevant network meta-analyses for the use of TIMS among participants with ulcerative colitis. The 2 most recent and comprehensive are summarized on these next few slides. We don't have time to go through them, but they are here for your reference and information. So we are going to skip over to the, I think it's slide 32, the discussion. Perfect. So just a few limitations to mention as you think about this evidence. First, there are frankly just a limited number of head-to-head studies in these 2 clinical conditions. No studies were powered for harm outcomes, particularly rare outcomes. Drug manufacturers did sponsor all of the eligible randomized trials. Most of the cohort studies used administrative or claims data, which may not be as valid or reliable as direct measurement, particularly for harms. And finally, we did not include reviews that were shorter than 12 weeks. We didn't include cohort studies with fewer than 1,000 participants. And we did not include data from conference abstracts, press releases, or studies published in languages other than English. So in conclusion, we found limited evidence for comparative effectiveness or harms of TIMS for Crohn's disease. For ulcerative colitis, we did find that vedolizumab is more effective than adalimumab with no difference in harms, but there's really limited evidence for other comparisons in ulcerative colitis. 13 studies are ongoing in this area. Some of them are head-to-head comparisons, some are studies of pipeline agents against placebo, but the results for all of them, none of them will have results available earlier than March 2021. And I think that's it. I'm happy to take any questions at this point on this particular review.
Hi, Layla. This is Nancy. Thank you so much for your review. I know you didn't spend a lot of time on the network meta-analyses, but I was just kind of curious looking at that table, like in the more thorough evaluation, vedolizumab kind of had more in favor than adalimumab, but in the network analysis table, it said like none for a lot of. What do you, based on your assessment of that network meta-analysis, which I know has a lot of flaws as well, what are your thoughts about that finding in that network meta-analysis? Thank you.
Yeah, that network meta-analysis was published before the trial that came out of the direct comparison of vedolizumab to adalimumab. So I would say the network analysis is useful information, but as soon as it's published, it's sort of obsolete when new studies come out. So I'd probably favor the direct head-to-head comparison if you're evaluating those two agents side by side, because it's a direct comparison versus this, which did not have the benefit of that study when it was conducted. Does that answer your question?
Yes, thank you very much.
Mm-hmm.
And this is Virginia Bucola, committee chair. Thanks again, Leela. That was a great presentation. Committee, are there any other questions for Leela? All right. So I believe we're going to move now to our stakeholders. We have two assigned stakeholders, Piao Ching representing Pfizer and Dr. Margaret Ullman representing AbbVie. If there are any other stakeholders present that I haven't listed, if you could please raise your hand for Lita Vasquez to be aware. And just as a reminder for the stakeholders, if you could state your name, and I'll have a little timer going for 3 minutes for each stakeholder. So we'll go now to Piao Ching.
Okay, just a second, and I will. Okay, Piao, you're unmuted.
Good morning. My name is Piao Ching. I'm a pharmacist with the Pfizer Medical Affairs team. I want to thank you for allowing me to provide medical information about tofacitinib, brand name Xeljanz, and Xeljanz XR in support of Pfizer's request to add tofacitinib to Apple Health preferred drug list. Tofacitinib is indicated for the treatment of adult patients with moderately to severely active ulcerative colitis, or UC, who have had an inadequate response or who are intolerant to TNF blockers. Use of tofacitinib in combination with biological therapies for UC or with potent immunosuppressants such as azathioprine and cyclosporine is not recommended. The recommended induction dose is 10 mg twice daily or 22 mg XR once daily for 8 weeks, and evaluate patients and transition to maintenance therapy depending on therapeutic response. If needed, continue 10 mg twice daily or 22 mg XR once daily for a maximum of 16 weeks. The recommended maintenance dose is 5 mg twice daily or 11 mg XR once daily. For patients with loss of response during maintenance treatment, a dosage of 10 mg twice daily or 22 mg XR once daily may be considered and limited to the shortest duration, with careful consideration of the benefits and risk for the individual patient. Tofacitinib includes a box warning for serious infection, mortality, malignancy, and thrombosis. While there are a number of available pharmacologic treatments, unmet medical need remains high, and new treatments are needed. A systematic literature review clinician survey found that TNF inhibitor primary failures and secondary failures with first-line TNF inhibitors was common for UC patients. In closing, UC continues to have a high unmet medical need in Apple Health population. To further manage this disease, we need alternative agents available after TNF inhibitor. Adding a medication with a novel mechanism of action and with oral administration will offer an additional treatment option for patients with UC in the Apple Health population. Based on the efficacy and safety of tofacitinib, we asked the committee to add Xeljanz and Xeljanz XR to Apple Health preferred drug list. I'll be happy to respond to any questions you may have. Thank you.
Thank you, Mr. Ching. This is Virginia Bucola, committee chair. If there are any questions from the committee for Mr. Ching? Okay, thank you very much. We'll move on to Dr. Margaret Ullman from AbbVie. And Dr. Ullman, you have 3 minutes once we get to you.
This is Lita. Margaret, I see that you are self-muted, so I've unmuted you. If you can unmute yourself. There you go.
Okay, great. Thank you. Hello, I'm Dr. Margaret Ullman from Medical Affairs at AbbVie. Thank you for the opportunity to speak with you. As you know, Humira has 10 FDA-approved indications. Today, we're examining the data that supported the approval of adalimumab, brand name Humira, for 3 of those indications: the treatment of adult Crohn's disease, pediatric Crohn's disease, and ulcerative colitis. Since this is only a short summary, please review the full prescribing information at www.rxabbvie.com for comprehensive safety and efficacy data. Humira, a TNF antagonist, is indicated to reduce the signs and symptoms and reducing and maintaining clinical remission in adult patients with moderately to severely active Crohn's disease who've had inadequate response to conventional therapy, or if they've lost response to or are intolerant to infliximab. The GAIN trial evaluated Humira induction therapy in adult Crohn's disease patients initially treated with infliximab and either lost response or discontinued its use as a result of intolerance. At week 4, 21% of Humira patients versus 7 of the placebo patients were in clinical remission. The CHARM trial evaluated maintenance of clinical remission in adults with moderately to severely active CD. Those who demonstrated a clinical response to Humira during a 4-week open-label phase were randomized to receive either Humira every week, every other week, or placebo. In the Humira every other week group, 40% of patients achieved clinical remission at week 26 versus 17% in the placebo group. The approved indication for Humira as a treatment of pediatric patients 6 to 17 years old with moderate to severe Crohn's disease followed the Imagine-1 study that evaluated its safety and efficacy with maintenance dosing regimens following induction. The efficacy and safety of Humira to treat adults with moderate to severe ulcerative colitis was evaluated in 2 clinical trials for the induction and maintenance of remission. At 52 weeks in the subgroup of patients with prior anti-TNF use, 10% of those in the Humira group were in clinical remission versus 3% in the placebo group. All TNF antagonists carry similar box warnings regarding serious infections, TB, and malignancies. Patients starting any anti-TNF, including Humira, should be screened for TB and carefully monitored for serious events. Humira has sustained efficacy, published long-term safety data, and a durable response in patients with Crohn's disease and ulcerative colitis. I respectfully urge the committee to maintain preferred status for Humira on the PDL for the people of Washington. Thanks so much for your time and attention. I'd be happy to answer any questions you might have.
Thanks, Dr. Ullman. This is Virginia Bucola, committee chair, just opening it up to the committee if there are any questions. Okay, I think we'll move on. I don't believe there are any other stakeholders for this classification, and we're going to.
I don't see any other stakeholders raising their hand.
And again, for the committee, we're just waiting until after all 3 sections have been reviewed to do a single motion, so we won't go to motion right now. Is that correct, Lita?
That is correct. I am opening up the next presentation.
So our next presentation will be on rheumatoid arthritis and ankylosing spondylitis, and we're welcoming Gerald Gartel. I practiced your name. Would you please say it for us?
Carolina.
Thank you very much and welcome. Please go ahead.
Yeah, thank you. So my name is Gerald Gartlina, and I will continue with the team's presentation, and my presentation is on rheumatoid arthritis and ankylosing spondylitis. Next slide. So this slide just provides an overview of my presentation today. I will start out with background, talk a little bit about the PICO criteria and the key questions. Then I would like to drop methods because they're basically the same as for Leila's presentation before on ulcerative colitis and Crohn's disease. And I will spend most of the time on findings and then end with the conclusions. Next slide. So we used several abbreviations in the slide set. They are all defined here on this slide. And they also cover frequently used outcomes in the report and in the slides. And I would like to point out 2 specific outcomes that are very commonly used for rheumatoid arthritis. And the first one is the ACR, the American College of Rheumatology response. It is a composite measure that includes the number of swollen joints, tender joints, and others. And an ACR 20 response, for example, is a 20% improvement in measures. An ACR 50 and 70 response are a 50 and 70% improvement. And in the report, we consider an ACR 20 and ACR 50 response as clinical improvement and an ACR 70 response as disease remission. And the second commonly used outcome is the DAS28, which is also a composite index for disease activity. And it combines an assessment of 28 joints with a global assessment of patient and laboratory markers. And the score goes from 2 to 10, where 10 is the worst. And a score of 2.6 is considered remission. And we also use this for remission in the report. And so these are the 2 most commonly used outcome measures that in the trials. Next slide. So I would just briefly walk you through some background. Next slide. So our report focused on 2 conditions, rheumatoid arthritis. Can we go back 1 slide, please? Yep, exactly. So rheumatoid arthritis and ankylosing spondylitis. Rheumatoid arthritis is a chronic inflammatory autoimmune disease of the synovial tissue that progressively erodes cartilage and bone, and that often can lead to a destruction of the joints and to disability. Ankylosing spondylitis is also a chronic inflammatory arthritis, but it is of the axial skeleton with a prominent involvement of the spine and the sacroiliac joints. And Leila has already introduced you to targeted immune modulators. Next slide. So this slide also provides a snapshot of the different TIM agents that we included for this update. Overall, we included 15 agents and 4 pipeline drugs. The agents in the gray boxes are approved for use in both rheumatoid arthritis and ankylosing spondylitis. The agents in the green boxes are approved for rheumatoid arthritis only. And the 4 white boxes show the pipeline drugs. Next slide. So this slide now presents our PICOS criteria. The populations were adults with rheumatoid arthritis or ankylosing spondylitis, and we considered any TIM agents that is FDA approved for these conditions or that is in the pipeline for approval. For comparators, we selected other TIM agents, and for pipeline drugs, we also included placebo or standard of care comparisons. For outcomes, as always, we were interested in health outcomes such as remission, clinical improvement, but also in adverse events or serious adverse events. And for study designs, we used the same criteria as for Crohn's disease. We selected randomized controlled trials. And in addition, for harms, we also selected cohort studies. Next slide. So our report addressed 4 key questions. Key question 1 was about the comparative effectiveness of the TIM agents. Key question 2 about comparative harms. Key question 3 was about subgroups. And key question 4 addressed the ongoing studies. Next slide. So these are the methods. I think we now can skip forward to slide 12. Yep, so findings and next slide. So overall, for this update, we included 23 new studies, and in total, the report now includes 76 studies, including 10 studies on pipeline drugs. 35 of these studies are RCTs, 41 are cohort studies, and the majority of the studies is on rheumatoid arthritis. Only 3 studies include data on ankylosing spondylitis. 24 studies addressed key question 1 on the comparative effectiveness of TIM's agents, and 62 studies addressed the comparative harms of TIM agents, and we did not find any studies for key question 3 on differences in subgroups. Next slide. So what were the findings? Next slide. I would like to start with rheumatoid arthritis, and the way we have structured the results on rheumatoid arthritis is that I will first talk about benefits and harms from RCTs on TIM agents as first-line treatments. Can we maybe go back one slide? Oh, there's one. Okay. And then about second-line treatment. So second-line treatments refers to patients who did not adequately respond to a TIM agent. And in the interest of time, I will present the results on the adverse events only if studies showed statistically significant differences. Next slide. So this slide is also an evidence map showing the number of RCTs and their methodological quality for the specific comparisons. So for first-line treatments, we had 15 RCTs in total, representing 11 different head-to-head comparisons and 2 comparisons in combination treatments with monotherapy. There are several things to note on this map. So first, most of the possible pairwise comparisons, as you can see from the empty cells, for most of those, we do not have any RCTs. So for most of the comparisons, there's no direct evidence. And second, the majority of trials are comparing a TIM agent to adalimumab. And all studies were sponsored by the industry and also had extensive industry involvement, which is then why overall we downgraded them to fair methodological quality. Next slide. So in the first comparison on abatacept versus adalimumab, clinical improvement and disease remission were reported by 1 RCT, which analyzed 646 participants. Clinical improvement and disease remission were similar between the 2 treatment groups, and we graded these findings as a low quality of evidence. For the second comparison, abatacept versus adalimumab, only 1 study reported similar clinical improvements. Can you click on the slide? Oh, sorry, they're separate. Okay. Thank you. So abatacept clinical. So abatacept versus adalimumab had similar clinical improvements and similar remission rates. But what I wanted to note here, abatacept actually had a significant lower incidence of serious adverse events than infliximab. And then the next comparison is abatacept versus infliximab. And we found similar improvements between abatacept and infliximab for ACR 50 and the clinical improvement, and there were also similar remission rates. And just for serious adverse events, there was a lower proportion for abatacept than infliximab, and the difference was 5% versus 12%. And we rated the quality of evidence for all of those as low quality of evidence. Next slide. For adalimumab versus baricitinib, we had one RCT that analyzed more than 1,300 participants, and this trial reported significantly fewer ACR20 responses for adalimumab than baricitinib. The difference was 61 versus 70%. And as mentioned in the beginning, an ACR20 response means a 20% reduction in symptoms on the American College of Rheumatology scale. Remission rates, however, were similar, but baricitinib had significantly higher incidence of serious adverse events compared with adalimumab. And the difference here was 8% versus 4%.
Sorry about that. Hang on just a second. Oops, I'm sorry. Hang on just a second.
Okay, then the next slide. So adalimumab versus certolizumab pegol. So we found one RCT with 915 participants. This was the Accelerate study. And this study reported similar response and remission rates between adalimumab and certolizumab pegol. We rated the evidence on clinical improvement as high quality. Next slide. Adalimumab versus etanercept. Can we go back to that again? Adalimumab versus etanercept, exactly. So we found two small RCTs with a total of only 167 participants that compared adalimumab with etanercept. And these two studies found no differences in clinical improvements. And we rated the quality of evidence as very low. Next slide. Adalimumab versus sarilumab. So there was also only one RCT that compared adalimumab to sarilumab. And in this study, adalimumab was less effective for clinical improvement, disease remission, and quality of life at 24 weeks. So for example, the ACR 50 response rate was 30% for adalimumab and 46% for sarilumab. We rated the quality of evidence as moderate for clinical improvement and quality of life, and as low for remission. Next slide. Adalimumab versus tocilizumab. So we identified two RCTs with a total of 369 participants. In terms of clinical improvement and remission, adalimumab was less effective than tocilizumab. The ACR 50 response, for example, was 28% versus 47%. And there is one caveat, however. In the larger one of these two trials, tocilizumab was administered at a higher dose than FDA approved. but quality of life was similar between the groups, and we rated all outcomes for this comparison as low. Next slide. Adalimumab versus tofacitinib. So there were no difference between groups in this in either clinical improvement or disease remission for the 3 RCTs that compared adalimumab to tofacitinib. The body of evidence includes more than 2,000 participants, and we rated this body of evidence as high quality of evidence. We also rated the quality of evidence as high for an RCT that compared adalimumab versus apaticitinib. So clinical improvement was lower with adalimumab than apaticitinib in ACR 50, which was 29% versus 45%. And similarly, significantly fewer participants achieved remission with adalimumab than apaticitinib. Next slide. For etanercept versus infliximab, we rated the quality of evidence as very low because this was a very small trial with only 32 participants, and it is really not possible to draw any conclusions from this study. And pretty much the same situation for etanercept versus tocilizumab, also a very small trial with only 43 participants. And results showed similar clinical improvements, but we can really not draw any conclusions from these 2 studies. Next slide. So in the report, we also included 2 RCTs that assessed combination treatments. So 2 targeted immune modulators combined with each other. One combined etanercept with anakinra, and the other one etanercept with abatacept. Overall results showed no or very limited additional benefits of a combination of 2 targeted immune modulators, but the serious adverse events were substantially higher in the combination group. So for example, 11% versus 3%. Next slide. So these were the comparisons on the first-line treatments for rheumatoid arthritis, and now moving on to the second-line treatments. So in patients who did not achieve adequate response with a first-line treatment. Here is an evidence map again. For second-line treatments, we found even fewer studies than for the first-line treatments. As you can see, we do not have any direct head-to-head comparisons for most of the drugs, and many of the available RCTs were of poor methodological quality. Next slide. So in the first comparison for second-line treatments, abatacept was compared to TNF-alpha inhibitors as a class. And the study showed similar treatment effects for clinical improvement or quality of life. And we rated the quality of evidence for these outcomes as low and very low. Next slide. Next. So similarly then, there was no difference between groups for the comparison between abatacept and rituximab. The quality of evidence was low for clinical improvement and quality of life. Next slide. One study assessed the comparison of abatacept versus secukinumab. The ACR 50 response rate was greater in participants treated with abatacept compared with those treated with secukinumab. It was 28% versus 17%. We rated the quality of evidence for this outcome as moderate. Next slide. And in the comparison of abatacept versus tocilizumab, one RCT with 132 participants showed similar clinical improvements between the two drugs, but adverse events actually were lower for abatacept than for tocilizumab, and also the proportion of serious adverse events was lower for abatacept than for tocilizumab. Next slide. We also found one pragmatic RCT that enrolled participants who had an inadequate response to a TNF-alpha inhibitor. And then the study randomized participants to another TNF-alpha inhibitor or a targeted immune modulator agent with a different mechanism of action. And results showed that switching to a TIM agent with a different mechanism can lead to a greater clinical improvement and to better remission rates than switching to another TNF-alpha inhibitor. And the remission rates here were 27% for the TIM agent with the different mechanism and 14% for switching to another TNF-alpha inhibitor. Next slide. And then finally, one small RCT showed that adding rituximab to a TNF-alpha inhibitor is more efficacious than maintaining the same TNF-alpha inhibitor to which participants did not respond adequately. So this was a small trial with 54 participants, and the result is not really surprising. Next slide. So I'm now moving on to the pipeline drugs for rheumatoid arthritis. Next slide. So three placebo-controlled trials evaluated filgotinib, which is a new Janus kinase inhibitor, and the trials included more than 1,300 participants, and all outcomes of interest improved significantly under filgotinib treatment compared with the placebo groups. Next slide. 5 placebo-controlled trials assessed peficitinib compared with placebo, and peficitinib is also another Janus kinase inhibitor. Clinical improvement and remission were also significantly greater under the peficitinib treatment than under placebo treatment. Next slide. We also found one head-to-head trial that compared peficitinib with etanercept. And in this study, etanercept was significantly superior regarding clinical improvement and remission compared with peficitinib. Next slide. We found another head-to-head trial that assessed a combination therapy of certolizumab pegol and bimekizumab, which is a new interleukin-17 inhibitor, and it compared the combination of these two with certolizumab pegol monotherapy. Can we go one back?
Sorry about that.
And one down. And one more down. Yeah, and one up again.
Oh, okay.
So the combination of certolizumab pegol plus bimekizumab versus certolizumab pegol monotherapy. And here the combination therapy led to higher proportions of response and remission than certolizumab pegol monotherapy. Next slide. So as I mentioned in the beginning, in addition to adverse events from RCTs, we also included data from observational studies. And I would like to briefly summarize these findings from the observational studies on the next slide. So overall, we included 41 cohort studies, which were mostly based on registry data. The majority of studies did not report any differences in serious adverse events such as mortality, malignancies, or cardiovascular events. There are two notable results, though, and several studies consistently indicated a higher risk for serious infections, opportunistic infections, tuberculosis, and herpes zoster for infliximab compared with other targeted other TNF-alpha inhibitors. And two studies reported a higher risk for gastrointestinal perforations for tocilizumab compared with TNF inhibitors. Next slide. So I'm now moving on to findings on ankylosing spondylitis. Next slide. So for down again, please. We're back up to the cohort studies. So going forward, yeah, exactly. So we found only one head-to-head trial for ankylosing spondylitis, which unfortunately also was of poor quality. Next slide. So this head-to-head study included 50 participants and compared etanercept to infliximab. The study reported smaller improvements for etanercept than infliximab, but we rated the quality as very low, and basically we cannot draw any solid conclusions from this study. Next slide. We also found one RCT on the pipeline drug filgotinib for ankylosing spondylitis, and this study found greater improvements for filgotinib than placebo for the treatment of ankylosing spondylitis. So filgotinib is still a pipeline drug. Next slide. We also summarize findings from two it says recent, from two network meta-analysis for TIM agents. They are the most recent network meta-analysis, but their searches actually go 3 and 4 years back, so they probably have been outdated by now. But you can find details on them in the report, and I will not present them here on these slides. Next slide. So the review limitations. So what are the limitations of our update? First, as you saw from the evidence map, for most comparisons, we do not have any direct head-to-head evidence. And when we have head-to-head studies, few comparisons actually were evaluated by more than 1 or 2 studies. And we are also missing long-term data on the effectiveness and safety. Drug manufacturers sponsored nearly all of the included RCTs, and most observational studies addressing harms were of retrospective design and based on national registries. And with these registry studies, the quality and completeness of the databases cannot really be determined. And then, of course, there are some inherent potential limitations of the methods. For example, we focused on English language studies only. Next slide. So we should skip the ongoing studies in the interest of time. So let's move on to slide 48, which are our conclusions. Next slide. So what are the conclusions? Well, we limit the conclusions here on evidence of high or moderate quality of evidence. And adalimumab appears to be less effective than baricitinib, sarilumab, and apodicitinib as first-line treatment for rheumatoid arthritis. Abatacept appears to be more effective than secukinumab as a second-line treatment for rheumatoid arthritis. We did not find any differences in the incidence of adverse events or serious adverse events. And the differences that we found in effectiveness and harms that we rated with low or very low quality of evidence, they must be interpreted cautiously. Next slide. So for ankylosing spondylitis, we cannot draw any firm conclusions. And this slide also concludes my presentation. Thank you very much for your attention. And if you have any questions, please go ahead.
This is Virginia Bucola, committee chair. Oh, good. I see Alex his camera's on, so just opening up to questions.
If I could just ask, I'm curious why adalimumab is so highly featured in the head-to-head trials. Is there a particular reason?
Yeah, I think the reason probably is that it was one of the first targeted immune modulators that came on the market. I think it was one of the first 3, and why then adalimumab and not etanercept, I don't really know, but yeah, but it is interesting that it has been used so often as a comparator.
Thank you.
And this is Virginia again. Any more questions from the committee for Gerald? I see Nancy.
Hi, Gerald. Thank you so much for your presentation. I know that you've done a lot of work in this drug class, and I had more of a question about the study with the 1,000 N of 1,000 patients, adalimumab with upadacitinib. Can't say the name, but in terms of that one appeared that upadacitinib is leaning more positively. Look, in your further research, I also know that this is a 24-week trial, so it's also short as well. Are there any additional things that we should be aware of? I mean, that does kind of seem to be kind of floating to the top.
Yeah.
But are there things that we should be concerned about or cautious about?
Well, you know, it is still a single study. It was funded by the maker of upadacitinib, but it was a well-conducted study, and it is a large sample size. So it had 1,629 participants. The methods were done well. So for us, it looked really good. One thing that is really always hard to judge, of course, is publication bias. So maybe there are other studies that we are not aware of that have never been published. But from what we had, this looked like a good, well done study with a large sample size, and that was also the reason why we graded it as high quality of evidence.
Great, thank you so much. I appreciate your thoughts and comments.
This is Virginia again. Thank you, Gerald. I appreciate your report. I'm assuming there are no more questions from the committee. Okay, we'll move on to stakeholders. The following 3 stakeholders are coming up next. We have Anthony Wheeler representing Eli Lilly. We have Pao Ching with Pfizer and Dr. Margaret Ullman with AbbVie. And if there are any other stakeholders, please raise your hand so that Lita Vasquez is aware. And each stakeholder, please state your name for the record, and you'll have 3 minutes for your words. Thank you.
This is Lita. I do not see Anthony Wheeler on the meeting, so I'm going to skip to Piao. I'm unmuting you now. Anthony, if you are on under a different name, if you can raise your hand. But Anthony, I'm sorry, Piao, you're up.
Okay. Thanks, Donna. Hello again, I'm Piao Ching with Pfizer Medical Affairs team. Thank you again for allowing me to provide medical information on tofacitinib, brand name Xeljanz and Xeljanz XR, for your consideration for Apple Health preferred drug list. Tofacitinib is indicated for the treatment of adult patients with moderately to severely active RA who have had an inadequate response or intolerance to methotrexate. It may be used as a monotherapy or in combination with methotrexate or other DMARDs. The recommended dose of Xeljanz is 5 mg twice daily. The recommended dose of Xeljanz XR is 11 mg once daily. A 12-month double-blind head-to-head non-inferiority randomized control trial was conducted to assess the comparative efficacy of tofacitinib 5 mg twice a day monotherapy, tofacitinib 5 mg twice a day plus methotrexate, and adalimumab 40 mg sub-Q every other week plus methotrexate for the treatment of RA in patients with previous inadequate response to methotrexate. This study was published in Lancet in June 2017. The primary efficacy endpoint was 50% improvement in ACR 50 at month 6. At this time point, ACR 15 response rate for tofacitinib 5 mg twice a day monotherapy was 38%. Tofacitinib 5 mg twice a day plus methotrexate was 46%, and adalimumab 40 mg every other week plus methotrexate was 44%. Using a non-inferiority margin of 13%, non-inferiority of the ACR 50 response at 6 months was shown for tofacitinib plus methotrexate versus adalimumab plus methotrexate, but not for tofacitinib monotherapy. The study authors noted no new safety signals with any of the treatments. A 2012 study of the Corona registry showed there was 71% to 85% non-responder to first and second anti-TNF biologic after 12 months. In addition, it was shown that a gradual reduction in efficacy was observed with subsequent therapy steps. Tofacitinib is included in the ACR RA treatment guideline post-methotrexate in established RA. According to the ACR guideline, if the disease activity remains moderate or high despite DMARD monotherapy, the recommendation is to use combination traditional DMARDs or add an anti-TNF or a non-TNF biologic or tofacitinib rather than continuing a DMARD monotherapy. In closing, adding a medication that is administered orally will offer an additional treatment option for patients with RA in Apple Health population. Based on the efficacy and safety of tofacitinib, we asked the committee to add Xeljanz and Xeljanz XR to Apple Health preferred drug list. Thank you for your attention, and I'm happy to take any questions.
Thank you, Piao. Are there any questions from the committee for Piao? Okay, I want to pause for a moment. I see that Anthony Wheeler is here, and I just want to pivot to Lita Abascus to see how if she would like Anthony to go next or move to Dr. Ullman.
Okay, I still don't see Anthony on the attendee list.
Under Lita, under the asker of the question, the name is Jill Somatic.
Oh, there we go. Okay. All right, Anthony?
Yep, here I am. Can you folks hear me okay?
Yes, thank you.
Yes.
All right, fantastic, thanks. Well, again, I'm Anthony Wheeler, and I'm an employee of Eli Lilly and Company, which manufactures Taltz. This is also known as ixekizumab, and it's an IL-17 inhibitor. And in this current category you're reviewing, I wanted to provide an update of a couple new indications that were approved for Taltz since your last review of this drug. This was originally approved for plaque psoriasis, but again, in this category, this drug was approved last summer for ankylosing spondylitis, and then was most recently approved just a couple weeks ago for non-radiographic axial spondyloarthritis. This disease is in the same family of spondyloarthropathies as ankylosing spondylitis, but just has some different diagnostic criteria. And this latter approval was based on a randomized controlled trial called the COAST-X study. And in this study, patients who received Taltz had a statistically superior to those who received placebo on the primary outcome measure, which was ACES-40, or a 40% improvement in spondyloarthritis symptoms. This was a 52-week study. The adverse event profile was similar to what was seen in previous trials of Taltz in psoriasis and other indications, but certainly see the prescribing information for all of the safety details on Taltz. So thanks for letting me provide this update, and I'm happy to try to answer any questions that you have.
Thanks, Anthony. This is Virginia. Committee, are there any questions for Anthony? Okay, thanks, Anthony. We'll move to Dr. Ullman.
Yes, hi, this is Dr. Margaret Ullman from Medical Affairs at AbbVie. Thank you for the opportunity to speak with you again. AbbVie now has 2 targeted immunomodulating medications available to treat patients with rheumatoid arthritis. I'd like to briefly review both Rinvoq and Humira and answer any questions you might have. Please see the full prescribing information at rxabbvie.com for comprehensive safety and efficacy data. Upadacitinib, with the brand name Rinvoq, is an oral JAK inhibitor indicated for the treatment of adults with moderately to severely active rheumatoid arthritis who've had an inadequate response to methotrexate or other non-biologic DMARDs. One extended release 15 milligram tablet is taken once daily with or without food. The phase 3 clinical program consisted of 5 studies in over 4,300 patients, and it's the medication that Gerald mentioned earlier. The individual trials included RA patients who were methotrexate naive, conventional DMARD inadequate responders, or biologic inadequate responders. Rinvoq met all primary and all secondary endpoints in all 5 trials, and significantly more patients achieved DAS28 remission and low disease activity versus controls in each trial. Rinvoq is the only approved JAK inhibitor to demonstrate inhibition of joint damage in its approved population of methotrexate IR patients. It is also the only targeted immunomodulator to show clinical superiority in RA treatment to Humira plus methotrexate. The most common adverse reactions in the upadacitinib trials were upper respiratory tract infections, nausea, cough, and fever. All anti-TNFs, including Humira, carry similar box warnings regarding serious infections, tuberculosis, and malignancies. JAK inhibitors also include a warning about thrombosis. Patients starting on any of these medications in this class should be screened for TB and carefully monitored for serious events. Humira has 10 currently approved indications that include 3 that we're discussing at this time. In adult patients with moderately to severely active rheumatoid arthritis, moderately to severely active polyarticular juvenile idiopathic arthritis, and in adult patients with active ankylosing spondylitis. Humira has long-standing safety data, years of on-market experience, and a well-defined published benefit-risk database with over 1 million patients treated. In summary, I respectfully urge the committee to maintain preferred status of Humira on the PDL and to add Rinvoq as a preferred treatment for Washington Medicaid patients. Thank you so much for your time. May I answer any questions for you?
This is Virginia, Committee Chair. Thank you, Dr. Ullman. Committee, are there any questions? Okay, and I believe there are no additional stakeholders.
This is Lita. I do not see anybody else raising their hand.
Okay, so we're scheduled for a break. I will go ahead. Why don't we go ahead and do a 10-minute break? It's 10:25 and we'll reconvene at 10:35.
Sounds good.
Okay, see everybody then.
Thank you.
This is Virginia, and it's 10:35. Hope everybody had a good break. We're going to go to Leila Kawadi, who's ready to present to us on plaque psoriasis and psoriatic arthritis. So over to you, Leila.
All right, thank you, Jenny. So I'm back again for the last of the three presentations on TIMS. This one is focused on persons with plaque psoriasis and psoriatic arthritis. Next slide. We have the same structure for the placentation. Let me go right into the background. So 2 slides forward, please. Plaque psoriasis is a chronic inflammatory disease that affects the skin, scalp, and nails. Its hallmark is erythrosquamous scaling skin lesions. Psoriatic arthritis is a chronic inflammatory arthritis that's associated with the skin disease psoriasis. And as we've already mentioned, we're focused here on targeted immune modulators, TIMs. The first TIM, alefacept, was FDA approved for psoriasis in 2003. The first TIM was approved, etanercept was approved for psoriatic arthritis in 2002. And of course, additional agents, including biosimilars, have been approved since then. Next slide. This slide provides a snapshot of the different TIM agents that we included in the update related to plaque psoriasis and psoriatic arthritis. There are 21 agents overall that cover 9 different mechanisms of action that are noted in the green boxes. The agents in the blue boxes are approved for use in both plaque psoriasis and psoriatic arthritis. The agents in the green boxes are approved only for plaque psoriasis, and the agents in the pale yellow boxes are approved for psoriatic arthritis. The agents in the white boxes are not yet approved for either condition, but I will note upadacitinib, a Janus kinase inhibitor, is FDA approved for rheumatoid arthritis, but for this particular report, we considered it a pipeline agent because it's not yet approved for psoriasis or psoriatic arthritis. Next slide. On this slide are the PICOS study selection criteria. Again, very similar to what you've already seen before. The difference obviously here is that we're focused on adults with plaque psoriasis or psoriatic arthritis. And again, we are focusing on comparative effectiveness, so mostly interested in head-to-head comparisons of TIM agents. Next slide are the key questions. Again, the first key question is around comparative effectiveness. The second key question is around comparative harms. And then the third key question is around variation in effectiveness or harms by subgroups. We will skip over the methods because they're largely the same as what we've already described to you. So we're going to skip ahead to the findings slide, which should be slide 12, I think. So here is an overview of the 38 total studies that we included. 20 of these studies were new to the update, and 18 were included from the previous report. 31 of the 38 studies are randomized trials, while 7 are cohort studies. 35 were fair methodological quality, while 3 were poor methodological quality. 30 of the 38 were conducted among participants with plaque psoriasis. 8 were conducted among participants with psoriatic arthritis. 32 of those studies were head-to-head TIM comparisons, while the other 6 were comparisons of pipeline TIM agents to placebo controls. And 26 of the 38 addressed comparative effectiveness, and then 32 addressed comparative harms, and we had no studies that addressed variation by subgroup. Next slide. So before we move into the specific findings, let me just provide you with a key to some of the abbreviations that are going to be encountered, similar to what Gerald mentioned. I want to just cover a few specific ones. Similar to Gerald's presentation, the studies of psoriatic arthritis largely rely on the ACR response as a measure of clinical improvement or disease remission, and we considered ACR 20 and 50 as clinical improvement and ACR 70 as disease remission, similar to what Gerald described for the RA review. In terms of other global improvement measures, the physician or investigator's global assessment is often reported, and that's a score of 0 or 1 generally reflects disease remission. And then the other two measures, the DLQI, the Dermatology Life Quality Index, and the PASI, or the Psoriasis Area and Severity Index, are specific to plaque psoriasis studies. The DLQI, a score of 0 or 1, means that the disease is essentially having no impact on their quality of life. And the PASI score is a measure of disease response. PASI 50 represents a 50% improvement in the score, 75% respectively. And so PASI 50 is clinical improvement, and PASI 75, 90, or 100 are essentially we consider disease remission. So these are the most common outcomes that you'll see in this body of literature. Next slide. So now we're going to move into the findings, and we'll start with plaque psoriasis first. So next slide. So this is an evidence map similar to what Gerald showed you for RA that shows all the possible TIM agents and possible pairwise comparisons. And the first thing you'll also notice, similar to RA, is that there's many cells here that are empty, meaning they don't. We don't have evidence for those direct comparisons. There were 21 trials total representing 14 different head-to-head comparisons, and the majority of trials, as you can see, are comparing TIM agents to either etanercept or to ustekinumab. There's only 3 comparisons of other TIM agents. So there's adalimumab versus guselkumab. There's risankizumab versus adalimumab, and then secukinumab versus guselkumab. All the other comparisons are either against etanercept or ustekinumab. All studies were sponsored by industry and had extensive industry involvement in the study design, execution, and reporting, which is why they were all downgraded to fair methodological quality. And then one study was poor quality because of insufficient blinding and switching of treatments that occurred during the study. All of these studies enrolled participants with moderate to severe psoriasis based on at least a 10% body surface area involvement and or PASI score, meaning a certain threshold PASI score, or both. And most studies were at least 3 to 6 months duration. In addition, some of the studies required participants to be naive to biological agents, but this was not universally the case. Next slide, please. Okay, I just need to readjust my screen here for a minute. Pardon me. Okay. So we have the same kind of format of presentation as you've seen before. In the top green box are the TIM agents being compared, the number of studies, the number of participants. So this first comparison is apremilast against etanercept, one study of 166 participants. And the bullets in the boxes below the green boxes are the outcomes that we evaluated with grade. I'll note that the full report does include other outcomes that may have been reported by the study, but for the presentation, we focused only on the outcomes that we graded. So for apremilast versus etanercept, there was no difference in the PASI 75 score at 16 weeks, and we graded that evidence as low quality. And in terms of quality of life, there was no differences in quality of life as measured by the DLQI, and that was also at 16 weeks, and that was also, we also graded that as low quality. Then the next comparison in the bottom of the slide is brodalumab versus ustekinumab. There were 2 RCTs that compared these 2 agents, and together combined analyzed over 3,000 participants. For disease remission, brodalumab resulted in a higher proportion of participants achieving a PASI 100 response at 12 weeks. And we graded this evidence as high quality. So the consistent findings across the 2 studies. Next slide. The next comparison is etanercept versus infliximab. This was evaluated in one small trial, 50 participants. This was a poor quality study. It had some issues with blinding and switching of treatments. Overall, fewer participants that were treated with etanercept achieved a PASI 75 response compared to infliximab. It was 35% versus 72% achieving a response, and we rated this evidence as very low quality. In this case, it was more for imprecision, but also for study limitations. The study also reported quality of life measures. They looked at a relative change in the SF-36 physical health and mental health component scores and reported no differences between the two agents. And we graded that evidence as very low quality. The next comparison was evaluated in two phase 3 multi-center trials. These trials compared etanercept with ixekizumab. Etanercept was less effective for achieving disease remission at 12 weeks as measured by the PASI 75 outcome, and it was also less effective for improving quality of life, and that was measured with the DLQI. And the absolute risk differences are provided on the slide. These studies, this evidence base, depending on the study, will report things in different ways. Some of them report things as absolute risk differences. Some of them report just the actual absolute incidences, and then we have to go ahead and calculate the absolute risk differences. So there are some of the outcomes are presented in slightly different ways across the different studies, but we try to tell you what is the direction of effect is whenever possible. Next slide. The next comparison is between etanercept and secukinumab. This was evaluated in one trial of over 1,000 participants. In this trial, they evaluated both a 300 milligram dosage of secukinumab and 150 milligram dose. And etanercept was less effective for achieving a PASI 75 response at 12 weeks. It was 44% versus 77% for the 300 milligram dose, and 44% versus 67% for the 150 milligram dose. And we rated this evidence as high quality. Similarly, etanercept was less effective for improving quality of life. The change in the mean DLQI score was 7.9 points for etanercept versus 10.4 points in the 300 milligram dose group and 9.7 points in the 150 milligram dose group. And this, we rated that evidence as moderate quality. The next comparison at the bottom of the slide is etanercept versus tildrakizumab. This was evaluated in one trial of just over 1,000 participants. 2 doses of tildrakizumab were evaluated, a 100 milligram or a 200 milligram dose, and etanercept was less effective for disease remission as measured by the PASI 75, both at 12 weeks and also at 28 weeks. Which is the 28-week data is not shown on the slide, but it was effective at both time points. And we rated the data at both those time points as high quality. For quality of life, etanercept was also less effective than both doses of tildrakizumab at both 12 weeks, which was we rated as a moderate quality of evidence, and at 28 weeks, which we rated as high quality evidence. Next slide. The next comparison is comparing etanercept to tofacitinib, and this study evaluated either a dose of 5 milligram or 10 milligram twice daily. And this is a good time to just remind you that at the time we did this review, tofacitinib was approved for psoriatic arthritis, but was not approved for plaque psoriasis. The FDA had declined to approve it in 2015. And had requested additional efficacy and long-term safety data. And we didn't identify any information to suggest the manufacturer has any plans to resubmit it for a plaque psoriasis indication, but this study did provide data on that comparison, so we've included it in the report. In terms of disease remission, etanercept was more effective than 5 milligram dosage based on the PASI 75 response. But it was no different compared to the tofacitinib 10 milligram dosage. We rated this evidence as moderate quality. Etanercept was also more effective than 5 milligram of tofacitinib for achieving a clinical improvement as measured by a PASI 50 response, but again was no different than the 10 milligram dose of tofacitinib. Etanercept was more effective than 5 milligram tofacitinib for improving quality of life, and this was based on a a DLQI change of 5 points or more, and that was moderate quality evidence. But again, there was no significant difference comparing etanercept to the 10 milligram dose of tofacitinib. Next slide. The next comparison is etanercept with ustekinumab. This was one study of just under 1,000 participants that evaluated a 45 milligram dose of ustekinumab, and a separate study arm evaluated a 90 milligram dose. Significantly fewer patients in the etanercept group achieved a PASI 75 response compared to either of the ustekinumab groups. We also, and that was those were the only outcomes that we graded for that particular comparison. The next comparison is guselkumab versus adalimumab, and there were 3 trials, RCTs, combined total of 1,600 or so patients. One study compared multiple doses and different dosing intervals of guselkumab, while the other 2 RCTs compared a dosage of 100 milligrams at weeks 0, 4, and 12 to standard doses of adalimumab. These studies included multiple primary and secondary endpoints, including physician's global assessment, a 0 or 1 response, a PASI 75 and 90 response. So for disease remission, we focused on the PGA 0 or 1 response, and the 100 milligram dosage regimen of guselkumab was more effective, with absolute risk differences ranging from as low as 16 to 28 percentage points across the 3 different trials. For quality of life, 2 of the trials reported using the DLQI 0 or 1 response, and the absolute risk differences there were 13 and 15 percentage points favoring guselkumab, but they were only statistically significant in 1 of the 2 trials. So same sort of magnitude of difference, but because of precision, significant in 1 but not the other. All 3 of the trials reported a mean change in the DLQI from baseline, and the absolute mean differences in change ranged from reductions of 0.6 points to a reduction of 1.7 points. Next slide. There was one trial that compared guselkumab to secukinumab, just over 1,000 participants. This particular study designated its primary endpoint as the PASI 90 response at 48 weeks. And based on this endpoint, guselkumab was more effective than secukinumab. You can see there was an 84% response rate for guselkumab compared to 70% for secukinumab. However, the study also reported disease remission as measured by PASI 75 at a combined endpoint of 12 and 48 weeks. And for this outcome, guselkumab was non-inferior but was not superior to guselkumab. So they did 2 different kinds of statistical testing. And guselkumab was also non-inferior when only looking at the 12-week outcomes. Guselkumab, based on the PASI 90 at 12 weeks, guselkumab had a lower numerical response, 69% compared to 76% for secukinumab, but study authors did not conduct any significance testing for that particular outcome at that particular time point. And that was per the study's analysis plan, which was somewhat hierarchical in order to control for type 1 error. Overall, we graded the disease remission outcomes as moderate quality. We downgraded it by a level because of the inconsistency of findings across the measures and the time points. Though, one might not view this as an inconsistency, but rather evidence of a better maintenance response, perhaps with guselkumab, but likely probably no difference in an induction response. The next comparison was ixekizumab versus ustekinumab. There was one trial of just over 300 participants. This study dosed ustekinumab based on weight, so it was 45 milligrams for people, I think it was less than 100 kilos, and 90 for people over 100 kilos. Ixekizumab was more effective for disease remission as measured by a PASI 90 at 12 weeks. It was 73% response versus 42% response, and also at 52 weeks, which data is not on the slide, but the response was 77% compared to 59%. Ixekizumab was also more effective for improving quality of life at 12 weeks. The DLQI 0 or 1 response was 61% versus 45%. And we graded both of these outcomes as moderate quality. Next slide. There was one trial comparing risankizumab to adalimumab. Risankizumab was, this was about 605 people. Risankizumab was more effective than adalimumab for disease remission at 16 weeks, and this is based on a PASI 90 response. Risankizumab was also more effective at improving quality of life based on the DLQI 0 or 1 response, and we graded both of those outcomes as moderate quality. And then for the comparison of risankizumab to ustekinumab, we identified 3 trials with a combined total of over 1,000 participants. Risankizumab was more effective than ustekinumab for disease remission at 12 to 16 weeks. So that was a time frame across the 3 trials. And that was as measured by a PASI 90 response. And the absolute risk differences across the 3 studies range from 28 to 37 percentage points higher response for risankizumab. Risankizumab was also more effective at improving quality of life as measured by the DLQI 0 or 1 response. And we graded both of these outcomes as high quality. Next slide. The next comparison is secukinumab versus ustekinumab. There are 2 trials, a combined total of over 1,700 participants. Ustekinumab was also a weight-based dosing in this study, 45 milligrams or 90 milligrams, depending on body weight. For disease remission, secukinumab was more effective as measured by a PASI 90 response. The absolute risk differences were 21 and 22 percentage points in the 2 studies, and that was at 16 weeks follow-up. Secukinumab was also more effective for achieving DLQI 0 or 1 response for quality of life, and we rated both of those outcomes as high quality. One of these 2 studies also reported outcomes at 52 weeks, and secukinumab remained superior to ustekinumab on all the measures of disease remission, clinical improvement, and quality of life at that further time point as well. All right, that kind of finishes up the comparative effectiveness. Now we're going to move into comparative harms for plaque psoriasis. So all of the 21 trials included for comparative effectiveness also reported on harms of TIM agents. In addition, we identified 5 cohort studies. Overall, in general, across the body of evidence, we observed very few differences in harms in the head-to-head comparisons. In the RCT body of evidence, between agent differences were typically in just one of several harm outcomes that may have been reported when differences were present at all. So in this presentation, I'm only going to describe findings where a statistically significant difference was observed in at least one of the harm outcomes in one study comparison, though I'm going to mention all of the harm outcomes for that comparison to put the one isolated finding into some context for you. So the next slide, the first comparison where there was a statistically significant difference was from a cohort study of over 100,000 participants that compared apremilast to adalimumab. In this particular study, there was a lower incidence of serious infection requiring hospitalization for apremilast compared to adalimumab, and the hazard ratio was 0.31, and as you can see, it excludes a null effect. We rated this particular finding as very low quality of evidence, and this study did not report any other harm findings. The next comparison is apremilast versus etanercept. This was reported in one trial of 166 participants. This study observed a lower incidence of adverse events for apremilast compared to etanercept. The risk ratio was 0.75. And again, excludes the null effects, so statistically significant. We rated this particular evidence as low quality. In this particular study, there was no significant difference in the incidence of serious adverse events, but that particular estimate was really too imprecise to draw a meaningful conclusion. Next slide. The next comparison where we had a statistically significant difference in harms was for etanercept versus adalimumab, and this was reported in 2 cohort studies. In the first larger cohort of over 100,000 participants, there was a lower incidence of serious infection requiring hospitalization for etanercept compared to adalimumab. The hazard ratio was 0.76, and we rated that evidence as very low quality. In the second cohort, which had over 7,000 participants, there was a lower incidence of serious adverse events for etanercept compared to adalimumab, and the incidence rate ratio there was 0.75, and we also rated that one as very low quality of evidence. In the comparison of etanercept versus tildrakizumab, which is in the bottom of the slide, this was from one trial of over 1,000 participants. This study reported harms over 2 different time periods. So they reported things over 0 to 12 weeks and then over 13 to 38 weeks. So there was a lower incidence of adverse events for the 100 milligram dose of tildrakizumab compared to etanercept across both time periods. But there was also a lower incidence for the 200 milligram dosage, but only during the second time period, the 13 to 28 weeks. There was no significant differences in serious adverse events for either dosage of tildrakizumab compared to etanercept in any of the time periods. Next slide. For the comparison of etanercept to ustekinumab, which was reported in one RCT and one cohort, there are no significant differences in overall adverse events or serious adverse events in the trial, which we rated as low quality of evidence. In the cohort study of over 7,000 participants, there was a higher incidence of serious adverse events for ustekinumab compared to etanercept. Incidence rate ratio is 2.4. In this observational study, participants were enrolled from a national dermatology registry in the UK. Participants were classified as to whether or not they had a clinical status that would make them eligible to participate in the clinical trial. And the incidence rate ratio 2.4 that I just mentioned is the estimate for participants with a clinical status that would make them not eligible to participate in clinical trials. So the thinking there is that those patients represent a more real-world sort of population than people who are eligible for clinical trials. There was a slightly higher incidence of adverse events in the participants who would have been eligible for clinical trials, but the magnitude was lower. So it was 1.3 incidence ratio among that more selected population. Next slide. Next is the comparison of infliximab versus adalimumab. It was reported in one cohort of over 100,000 participants. In this study, a higher incidence of serious infection requiring hospitalization was observed for infliximab compared to adalimumab. Hazard ratio was 1.9, and we assessed that evidence as low quality. The next compared risankizumab versus ustekinumab, and that was reported in 3 trials of over 1,000 participants. One of those trials reported no significant difference in any harms. The other 2 reported harms over 2 different time periods. There were some slight differences in harms between the two periods, but there was no sort of consistent patterns for any single time period or harm. Next slide. And finally, for the comparison of ustekinumab versus adalimumab, there were 2 cohort studies. In one study of over 100,000 participants, there was no significant differences in serious infection requiring hospitalization, and that evidence was re-rated as very low quality. In the other cohort of over 7,000 participants, there was a higher incidence of serious adverse events for ustekinumab. Incidence rate ratio was 1.2, and we rated that evidence as very low quality. Again, this last finding is from the study comparing estimates from populations that either would be eligible or ineligible to participate in clinical trials. All right. Next slide, we did summarize some findings from 3 recent network meta-analyses for TIM agents for plaque psoriasis. There's some summary slides in the back of the deck for your reference, and the details of that are in the full report if you're interested in that. We are going to, for the sake of time, not go through the slides on pipeline agents, but there were 3 pipeline agents, and the results for those are all summarized in the slide deck and in the full report. So we are going to go ahead and skip to slide 33. Okay, and so we're going to move now into psoriatic arthritis. Next slide. This first slide is the evidence map for comparative effectiveness You can see we only identified a total of 5 trials. 2 were poor methodological quality and 3 were fair. All but one of the comparisons are comparing a TIM agent to adalimumab. The 5th study compares ustekinumab to tumor necrosis factor alpha inhibitors generally, like the class. The specific one that was selected by participants and their providers, so this was not a blinded study, and is one of the reasons we assessed it as poor quality. So next slide, we'll quickly go through the psoriatic arthritis comparison. So the first comparison was a trial of about 100 participants comparing adalimumab to etanercept. This study reported similar ACR20 responses at 1 year for all the agents. So adalimumab, etanercept, and infliximab all had similar responses at 1 year. However, the study did not really conduct statistical significance testing because this is not an aim of the study. And then on the bottom of the slide is the comparison of adalimumab to ixekizumab. This was a trial of over 400 participants. This study also reported similar ACR responses among participants. Again, also did not conduct any statistical significance testing, as the study also had a placebo arm, and the main point of the study was to compare ixekizumab to placebo. So they did not do testing for the active comparator arm. This study also reported a numerically lower skin disease remission response compared for adalimumab compared to ixekizumab, and also reported on ixekizumab dosing every 2 weeks and every 4 weeks. So there were multiple study arms in this particular study. Next slide. We compared one study that compared adalimumab. Actually, I think. One study compared looking at 422 participants, compared adalimumab to 2 doses of tofacitinib, 10 milligrams twice daily, or 5 milligrams twice daily. The ACR20 response was 60% for adalimumab compared to 70% for the 10 milligram dosage and 68% for the 5 milligram dosage. This study also did not conduct statistical significance testing. We graded that evidence as low quality. They also reported skin disease remission using the PASI 75, and it was 56% for adalimumab compared to 67% for the 10 milligram dose and 56% for the 5 milligram dose. Again, no statistical significance testing was conducted by the authors. We also graded that evidence as low quality. And then finally, for quality of life, study authors reported a change in SFPCS scores. They're all in the same neighborhood across the doses, but there was no statistical significance testing reported. And so we graded all of this evidence as low quality. All right. I think the next slide, you can go ahead to the next slide. This is comparing adalimumab to remtolumab, which is a pipeline agent. So I think we'll go ahead and skip that. Let's move to slide 38, which is the final comparison of ustekinumab versus a class, TNF alpha inhibitors. This was only in 47 persons. And these were all participants with active enthesitis, which is inflammation where the tendons and ligaments insert into the bone. So it's sort of a subset of people with psoriatic arthritis. And participants were randomized to, who were randomized to the TNF alpha arm selected their preferred agent based on the route and the frequency of administration that they preferred. In terms of both enthesitis and skin disease remission, ustekinumab was more effective at 24 weeks, but there was no difference in arthritis remission. And we graded the remission outcomes as very low quality evidence. And in terms of quality of life, ustekinumab was more effective as measured by the SF36 physical health component score, but there was no difference in the mental health component score. Next slide. In terms of comparative harms, 4 of the 5 trials also reported comparative harms for psoriatic arthritis. We did not identify any cohort studies, and there was only one significant difference in harms between agents, and that's described on the next slide. This was the trial comparing, if you go to the next slide, that compared adalimumab to etanercept and infliximab, 100 participants. There was a lower incidence of adverse events with adalimumab compared to etanercept. The relative risk, or the risk ratio, is 0.38. There was also a lower incidence with adalimumab compared to infliximab. That risk ratio was 0.23. The adverse events, incidence of adverse events was higher for infliximab compared to etanercept, and that risk ratio was 1.6. And we rated all these outcomes as very low evidence. All right. The next part of the slide deck is again on pipeline agents. So we're going to skip those and go to slide 43, which is just a summary of the limitations. Many of these are similar to what Gerald and I have presented earlier. There's quite a bit of direct evidence that we're lacking for many of the potential TIM comparisons, head-to-head comparisons. There's limited long-term and safety data, efficacy and safety data. Most studies aren't powered for those harm outcomes. Some of the cohort studies use claim data, which we've talked about those limitations. And again, manufacturers sponsored nearly all of the trials, and the same kind of methodological limits to the review, including, mostly reliance on English language studies and no data from conference abstracts or press releases. All right, we're going to skip over ongoing studies and go to conclusions, which should be slide 47. The largest body of comparative direct evidence in terms of plaque psoriasis is for etanercept and ustekinumab compared to the other TIM agents. And this is for disease remission outcomes. So high quality evidence suggests that etanercept is less effective than ixekizumab, secukinumab, and tildrakizumab. High quality evidence also suggests that ustekinumab is less effective than brodalumab, risankizumab, and then moderate quality suggests it may also be less effective than ixekizumab. Next slide. For the other TIM comparisons in terms of disease remission outcomes, high quality evidence suggests that adalimumab is less effective than guselkumab, and moderate quality evidence suggests that it's also less effective than risankizumab. Moderate quality evidence suggests that guselkumab is more effective than secukinumab for maintenance therapy. Few differences in harms among the TIM agents were observed based on very low to moderate quality evidence. And then lastly, in conclusion, for psoriatic arthritis, there's limited head-to-head comparisons available, but based on low quality evidence, ixekizumab, tofacitinib, and remtolumab may be more effective than adalimumab for disease remission with no difference in harms. And that's all in terms of the slide deck. I'm happy to entertain any questions at this point.
This is Virginia, just making sure that there aren't any questions from the committee for Leila.
Hi, Leila. Thank you again for this great work. I know it's a lot of information and trying to pare it down for us. Thank you. Just a little bit of background. We received a letter and from a provider in eastern state of Washington, and one of the comments or points that he made was about by the time they get to the preferred drug, they no longer see the potential efficacy that the patient could have obtained. And for me, this kind of triggered a question about disease severity and applicability of the patient populations. And I know that you really looked at a huge amount of information and data. Could you give us a little bit of insight into like maybe approximately what % of patients were naive to these therapies, or had they already have tried like one or two agents before, especially with some of these head-to-head studies?
Yeah, that's a great question. So what I can tell you is that the patient populations that were enrolled in these trials, the criteria for enrollment was remarkably similar across the study. So most of them required moderate to severe psoriasis, and now I'm really focusing on the plaque psoriasis component. Moderate to severe psoriasis, at least 10% body surface area involvement, and a duration of disease, usually a minimum of 4 to 6 months. So the patient enrollment criteria was fairly standard. Where the studies differed was in terms of whether they required patients to be biologically naive. And I would say I don't have all that data right at my fingertips, but I would say most of them probably did not. Some of them did require participants to be biologically naive, but most, I would say, over half did not require people to be biologically naive. Now, I need to go back and dive in if you wanted more specific numbers, because I just don't recall them off the top of my head. But that was where studies varied, was whether they required people to be naive or not.
Great, thank you so much. I appreciate it.
Mm-hmm.
And this is Virginia again, just checking in. Any more questions? Okay. Leila, thank you very much. All right, we have 4 stakeholders. We have Anthony Wheeler with Eli Lilly. We have Piao Ching with Pfizer, Carrie Johnson with Amgen, and Dr. Margaret Ullman with AbbVie. If there are any other stakeholders present that I haven't listed, please raise your hand and let Lita know.
Okay, Anthony, I've unmuted you.
Okay, great. Thanks so much. Again, I'm Anthony Wheeler, and I'm an employee of Eli Lilly and Company, which manufactures ixekizumab. This is also known as Taltz. It is an IL-17 inhibitor. And I'll provide just a short research update on a couple of the indications that are being reviewed in this report. The original indication that Taltz received was plaque psoriasis, and it was approved a couple years ago also for psoriatic arthritis. The first research update is the completion of a study called IXORA-R. This was a randomized controlled trial that compared Taltz with Tremfya, it's also known as gazelkumab, in participants with plaque psoriasis. In this study, Taltz showed superiority to Tremfya on the primary outcome measure, which was PASI 100, or complete clearance of skin lesions. Then the other study was known as Spirit Head-to-Head. This was also a randomized controlled trial, and it compared Taltz with Humira or adalimumab in participants with psoriatic arthritis. In this study, Taltz demonstrated superiority to Humira on the primary outcome measure, which was the simultaneous achievement of ACR 50, or a 50% reduction in disease activity, and PASI 100, or complete skin clearance. So thanks again for letting me provide these updates, and do see the package insert for all the safety details on Taltz. I'm happy to try to answer any questions that you may have.
This is Virginia. Thanks, Anthony. Any questions for Anthony from the committee? Okay, thanks very much. We'll move to Piao Ching.
Hello again, this is Piao Ching with the Pfizer Medical Affairs team. Thank you again for allowing me to provide this comment pertaining to Inflectra, a biosimilar to Remicade. The introduction of biosimilars provide patients and prescriber treatment options, which may expand access by offering potentially less costly alternatives for biologic medicines. The FDA approved Inflectra in April 2016. Inflectra is a tumor necrosis factor, or TNF blocker, indicated for active psoriatic arthritis and chronic severe plaque psoriasis in patients who are candidates for systemic therapy and when other systemic therapies are medically less appropriate. Inflectra is also indicated for moderately to severely active RA in conjunction with methotrexate, active ankylosing spondylitis, and patients who had an inadequate response to conventional therapy for moderately to severely active Crohn's disease and ulcerative colitis. Extensive comparison of the functional, physiochemical, protein, and higher order structure attributes match the pre-specified criteria for analytical similarity of Inflectra, US licensed Remicade, and EU approved Remicade. In a 3-way pharmacokinetic or PK study in healthy subjects, the pairwise comparisons of Inflectra, US licensed Remicade, and EU approved Remicade met the pre-specified acceptance criteria for similarity. The 90% confidence intervals for the geometric means ratio, or GMR, of AUC and Cmax were contained within the 80% to 125% similarity range. Comprised of 803 subjects, the safety database identified no new safety signals. Inflectra and the comparator products had similar incidences of treatment emergent adverse events, serious adverse events, and adverse events leading to discontinuation, infection, infusion-related reactions, and anaphylaxis. The FDA was provided with extensive data to address the scientific consideration for extrapolation to conditions of use not clinically studied in the Inflectra development program, thereby granting licensure for the 7 indications described previously. Inflectra is labeled with the same warnings and precautions as Remicade, including the black box warning for serious infections and malignancy. In closing, I would like to thank the committee for allowing me to present this testimonial, and I'll be happy to address any questions. Thank you.
Thank you, Piao. This is Virginia. Right, any questions for Piao? Okay, before we move to the next speaker, I just want to add that there looks like there's a 5th stakeholder, Shirley Quach, representing Novartis, and I'll add them to the end of the list. Up next, we'll go to Carrie Johnson, representing Amgen.
Hi, this is Carrie Johnson. I'm a pharmacist with medical affairs at Amgen. Thank you for the opportunity to speak in support of Otezla, or apremilast. Apremilast was FDA approved in 2014 for the treatment of adult patients with active psoriatic arthritis, adult patients with moderate to severe psoriasis who are candidates for phototherapy or systemic therapy, and now in July 2019 for the treatment of adult patients with oral ulcers associated with Behçet's disease. Warnings and precautions include diarrhea, nausea, and vomiting, depression, weight decrease, and drug interactions. Please see the full prescribing information at amgen.com for further information. Important reminders: apremilast is not a biologic, and recent published guidelines place it in a separate category. It is an oral small molecule that works intracellularly to inhibit phosphodiesterase 4. This reduces the cell's production of pro-inflammatory cytokines and increases production of anti-inflammatory cytokines. Importantly, apremilast has no black box warnings and no requirement for screening or laboratory monitoring. As an oral small molecule, it does not induce the production of anti-drug antibodies. I'm going to provide 4 updates, 2 label updates in this past year and 2 other updates. In July 2019, apremilast became the first FDA-approved therapy for the treatment of oral ulcers in adult patients with Behçet's disease. This is a rare chronic multisystem inflammatory disease that affects approximately 5 in 100,000 people in the US. Oral ulcers occur in more than 98% of these patients and can be painful and significantly affect quality of life. In the phase 3 RELIEF study, apremilast demonstrated significant improvement versus placebo at week 12 in the number and pain of oral ulcers associated with Behçet's disease. Most commonly reported adverse events in this trial included diarrhea, nausea, headache, and upper respiratory tract infection. Second label update in the past year: scalp psoriasis occurs in over 90% of patients with psoriasis and is considered a difficult to treat aspect of psoriasis. In the phase 3 STYLE study, apremilast demonstrated significantly greater improvement in scalp psoriasis, scalp and whole body itch, and quality of life versus placebo at week 16, with improvements continuing out to 32 weeks. Most common adverse events were diarrhea, nausea, headache, and vomiting. These data are fully published and were added to the label in April of this year. Long-term data, now published out to 5 years in psoriatic arthritis and over 3 years in psoriasis, shows no increase in incidence or severity of adverse events and no new safety signals over time. Recently published claims analyses demonstrate that biologic-naive patients with psoriatic arthritis who initiated apremilast had similar switch rates to biologic users and significantly lower healthcare costs regardless of treatment switching. In summary, apremilast is not a biologic and is placed in a separate category in recent guidelines. Apremilast does not have a black box warning and has no requirement for screening or laboratory monitoring. As an oral small molecule, it does not induce production of anti-drug antibodies. Apremilast represents an important oral, non-biologic option for your adult patients with moderate to severe psoriasis, active psoriatic arthritis, and now for treatment of oral ulcers associated with Behçet's disease. Thank you for your time, and I'll take any questions.
This is Virginia. Thank you, Carrie. Any questions from the committee for Carrie? Okay, moving to Dr. Margaret Ullman with AbbVie.
Hi, this is Dr. Margaret Ullman again. Thank you very much for today's opportunity to speak with you. AbbVie now has 2 targeted immunomodulating medications available to treat patients with psoriasis. I'd like to briefly review Skyrizi and Humira and answer any questions you might have. Please see the full prescribing information at rxabbvie.com for comprehensive safety and efficacy data. Risankizumab, with brand name Skyrizi, is an IL-23 inhibitor indicated for the treatment of moderate to severe plaque psoriasis in adults and is given as a subcutaneous injection at week 0, 4, and then every 12 weeks, which means 4 doses per year for maintenance treatment. The phase 3 clinical program was conducted in 4 trials with over 2,000 patients, and it met all primary and all ranked secondary endpoints in all trials. Skyrizi showed superior efficacy to Stelara, ustekinumab, in both PASI 90 and PASI 100 responses at week 16 and at week 52. After 2 doses, 75% of Skyrizi patients had at least a 90% reduction in their PASI score, and that proportion of patients increased to 83% after 1 year of treatment. Skyrizi also showed significance versus Humira in PASI 90 at week 16, and after a switch from Humira intermediate responders. In clinical studies, treatment for 1 year provided complete skin clearance to over 50% of patients. The incidence of adverse reactions in the integrated analysis was similar for Skyrizi, Humira, and Stelara through week 16 and in the long term. There were no unexpected safety findings, and there are no contraindications to Skyrizi treatment. Patients starting Skyrizi or Humira should be screened for tuberculosis and carefully monitored for serious infections or other adverse events. Humira has 10 currently approved indications that include both reducing the signs and symptoms, inhibiting the progression of structural damage, and improving physical function in adult patients with active psoriatic arthritis, and as a treatment for adult patients with moderate to severe plaque psoriasis in patients who are candidates for systemic therapy. With long-standing safety data, 71 global clinical trials, 14 years of on-market experience, and over 1 million patients exposed, Humira has a well-defined published benefit-risk database. In summary, I respectfully urge the committee to maintain preferred status of Humira on the PDL and to add Skyrizi as a preferred psoriasis treatment for Washington Medicaid patients. Thank you so much. I'd be happy to answer any of your questions.
This is Virginia. Thanks, Dr. Ullman. Pardon me. Any questions from the committee? Okay. We have one final stakeholder, and that is Shirley Quach with Novartis. Shirley, you have 3 minutes to speak.
Great. Thank you. Hi, my name is Shirley Quach, and I am a regional account MSL for the Northwest at Novartis. I just wanted to first thank you for all your work and effort in your review of the drug class, and thank you for this time for me to provide comment and adding me at the last minute. I wanted to provide some comments and some updates on Cosentyx. So Cosentyx is a fully human monoclonal antibody which selectively targets IL-17A, which is a key inflammatory cytokine that affects both the adaptive and innate immune system. I want to provide an update to one of the slides or clarify because Cosentyx is FDA approved for plaque psoriasis, but also psoriatic arthritis and ankylosing spondylitis. On Tuesday, we did have an additional approval for Cosentyx, and that was in non-radiographic axial spondyloarthritis, which is a part of the axial spondyloarthritis disease spectrum, and this is based on the phase 3 PREVENT trial. Cosentyx has also demonstrated consistent long-term efficacy and safety with 5-year data in patients with psoriasis, psoriatic arthritis, and ankylosing spondylitis, demonstrating Cosentyx's consistent efficacy and durability. It also improves all GRAPPA domains of psoriatic disease, such as axial disease, enthesitis, dactylitis, skin and nails, which some of these are considered hard to treat domains. And Cosentyx has demonstrated superior efficacy in psoriatic plaque psoriasis. We also have additional head-to-head trials that are ongoing in psoriatic arthritis and ankylosing spondylitis. There has been no additional safety concerns that have popped up in our long-term studies. I also wanted to address one of the questions that was asked regarding the patients that were treatment naive in some of these studies. For Cosentyx pivotal or trials, they vary across trials, but there are treatment naive patients up to 50% in each of our trials. And I can provide that information if you're interested. Thank you for your time. And if there are any questions, I'd be happy to answer it. Thank you.
Thank you, Shirley. Any questions from the committee? All right, so I think we need to move to this motion and get ready for some big word pronunciations.
This is Lita. I'm opening it up.
Okay, thank you.
I'll scroll down for you to view the last motion.
And I'm going to go ahead and turn my camera off again. I'll just open it up to the committee, knowing I can't see any of you. So if there's more time needed or we're moving too quickly, please let me know.
It's Alex Park here. Can I ask a question? Can you hear me?
Yes.
So
yes.
It's been a couple months since we've had one of these meetings, so I just wanted to confirm something. The language here seems a little different than what we've had before. I seem to recall we usually say something like it's safe and efficacious, but this prior motion only seems to say efficacious, and I can't remember if there was a reasoning for that or if I'm mixing up our procedures here between the P&T and the DUR motions.
This is Lita Vasquez. You're right. The motions do usually say safe and efficacious. Donna, do you recall why safe was left out of this one?
Hi, this is Ryan Pistoresi. I'm going to look at some previous motions and just see if it was omitted or if we've left it off for a little while.
Yeah, this is Donna. I think one of the reasons why you have left the safe off of the out of the motion for other drug classes that I recall is whether there might be a side effect or an adverse reaction that is potentially an unsafe adverse reaction. And so instead of saying that they're unsafe, which would go against an FDA approval that has said it's safe and effective, you have just omitted the word safe. I don't recall the reason for this particular drug class, but I recall that in general as an option that you have chosen to do as a committee.
Thanks for reminding us about that. I think that makes sense, given the number of black box warnings in this particular class.
Okay.
Yeah, thank you. I think that's resonating with me as well. I had a question. I know the presentations were done in different chunks. You know, for like ulcerative colitis, rheumatoid arthritis, plaque psoriasis, psoriatic arthritis. And some of the results obviously differed depending on the different disease states. Did we want to, this is a question for the, I don't know, for healthcare authority and committee members, like follow along with the how it was presented with like ulcerative colitis, rheumatoid arthritis, plaque psoriasis, psoriatic arthritis. That was just a question I had, because there's a lot of differences when we looked at, or when the evidence was presented.
So this is Ryan Pistoresi. So this is the first time that we've had DERP split it out by the different indication. And that's primarily because as this class has so many different drugs and so many different disease states, that it was very burdensome to do the whole report at once. And actually, when we commissioned this report from DERP, they actually split it out and delivered it to the states across several months. Which is why it was presented as these separate presentations, which then allowed us as states to review, kind of just this focused area at a time. So on the Washington PDL, it is arranged as, one big drug class, and this is really the first time that we've actually reviewed it by each kind of individual subsection.
This is Virginia, committee chair. I do recall that Lita, had been offered that if this motion felt too broad, we could break it into 3 smaller motions. And I don't know if that addresses any of your questions or wondering, Nancy.
Yeah, I guess that would be a question that I have for other committee members as well. Do you feel, and also I don't know how that impacts the healthcare authority, but because it's gotten so large, and my guess is it will continue to get larger, especially with pipeline medicines. I'm not sure we can clump them together anymore, but that I'd like some input from other committee members and healthcare authority staff as well.
So this is Ryan. Oh, go ahead, Donna. Near you.
What I was going to say was that this is a class that's going to be extremely difficult to lump or split by disease state. The reason being is that there are some that are indicated for multiple disease states and then others that are indicated for only one. And I think with the pipeline, that indication list is going to grow. So I think it is challenging to split them out, other than to, if you did, it would be by potentially their mechanism of action, which is also challenging because there's multiple indications that they treat. So I think it's best to leave them lumped together and then make recommendations to the committee based on making sure that there's one preferred product for each, FDA indication.
Hi, this is Ryan Pistoresi at Healthcare Authority. Just to follow up to Donna's point, when we do look at the cost analysis that follows this step in selecting the preferred drugs, we do look at it for the different indications and make sure that we are meeting your motion for the rheumatoid arthritis, ankylosing spondylitis, Crohn's disease, and so on and so forth. So it may make more sense to keep it at one large drug class for the PDL rather than split it out by indication, because then we would have repeating drugs across different disease states. And I was able to look back at previous motions. The last time that we used safe and efficacious in the motion for the TIMS was in 2008. And so since then, it's just been efficacious. So for the last decade, we have only used efficacious.
This is Alex Park. Nancy, I share your concern. It is kind of an unwieldy group, including their pronunciations. I mean, just thinking about how this, it's about patients in the end. And so would you feel more comfortable if we added some indications in the list if you feel like there's something we missed?
Now that I got some additional background from Donna and Ryan, I can see the points. So there's an echo. If we say that, in terms of like the FDA indications and in terms of like the clinical studies, maybe we can, I don't know how we would add wording to say something of the sort that based on the current literature for these individual disease states, that like Donna was saying, that there would be like a preferred agent based on current evidence that DERP has provided. I don't know if that kind of meets both aspects. I'm not sure if I'm articulating myself very well, but I understand, Donna and Ryan, your point, and that makes sense to me, to say that, you know, they're approved because they're efficacious, they're FDA approved, but then maybe adding something in addition to kind of, I guess, pull out that there are some differences in the evidence base for different disease states within, I don't know.
Great, so this is Ryan again. I think what we may want to do is we can start by copying the old motion over into where the new motion is, and then split out the list of the drugs and the PDL recommendation into two sections. So that way you're able to see how we may want to edit it to kind of accommodate what we saw in the DERP reports today.
This is Lita. So what I'm doing is there's two drugs that were not continued into this review, and they're in black here, Lefacept and canakinumab. So I'm just going to take those out and I'm going to put these new drugs in.
Donna, this is Nancy. I'm trying to remember exactly how you said it, but I liked in terms of phrasing it where we'd have like a preferred within the disease state.
Yeah, I mean, it's really challenging to have a preferred drug. I mean, I think we need a preferred drug that covers each of the indications, not necessarily a preferred drug for psoriasis. It's challenging to say one drug is preferred for this condition, but it's not preferred for that condition. It's either preferred or it's not. Or it's first line or it's, you know, second line. So I guess the question is, it's like some of the newer drugs, do you feel like they are superior enough to call them out as must be preferred, or maybe in special populations they should be preferred? That's another avenue to go down. That way, you know, when it's a special population, we could put limits on it or put it on prior authorization to make sure that only that particular special population received access to that drug. So it would be preferred, but on prior authorization. With Medicaid, we can do that through an expedited authorization process, so it's not as onerous as a formal prior authorization. But that allows access to those medications for those individuals that have that need without opening it up broadly to the entire population to be used for other indications where there might be less costly alternatives that are equally effective.
I would be in favor of that. And I don't know if other committee members feel similarly.
I have a different issue. So, you know, within the preferred drug list, are we calling out route at all? Because if you look at this list of drugs, some of them are given sub-Q, some are oral, some are infusion. How does that work too? Is one preferred over the other based on route, or are they all kind of clumped together? Or is it?
So this is Donna. Sorry, Susan. They are all clumped together right now. If you look at the bottom, you know, at the bottom of the motion, it says it should include like a self-administered agent. You could alter that and say it needs to be a self-administered, IV, oral, whichever routes that you think are important for us to have as preferred options. And then that will, you know, help us guide our PDL selection. I know with the triptan class, you have said all routes of administration must be, there must be a preferred product for each route of administration.
Okay. Well, I had to say, yeah, I would think that we would want to have a preferred. And I know all these indications, there's not always a sub-Q option or a infusion option, but if some patients aren't able to take oral, then it's nice to know that they could have an option. And then I agree too that I feel like, I don't know if we should necessarily break out this class based on indication, just because there's so much overlap.
Yes, so this is Nancy. I agree with what the input from HCA, and wanted to bring back the recommendation of kind of, Donna, what you just said prior, about maybe expedited review, and just to make sure that patients do have access to some of these, especially, I mean, we know that there's limited information and data, and the strength of the evidence varies. But for, like rheumatoid arthritis, it looks like one of the medications is a little bit, it's leaning towards one over the others. But there's also limitations there as well. So I just kind of wanted to bring it to the committee for their thoughts.
This is Virginia, and I just wanted to say that I'm in agreement with Nancy on that suggestion. In terms of wordsmithing, I might turn it back to our experts.
And this is Donna. So Nancy, did you have actual drugs in mind that you felt should be called out for these special populations? I think that the best way to do that is to specifically name those in the motion and then identify the special population that you feel that they should be preferred for.
So I'm going to, this is Nancy, I'm going to fall back on what the DERP report. So for like ulcerative colitis, is it the vedolizumab appeared to be more in favor for ulcerative colitis. And please, if I get anything incorrect, feel free to correct me. For rheumatoid arthritis, it appears that, is it up?
Hey, Nancy, this is Lita. Sorry to interrupt. So do you want to say that vedolizumab has to be preferred for I'm sorry, which type?
Ulcerative colitis. I don't know, Donna. I'll also defer to your wording recommendations.
And this is Donna. I mean, without having the slides, I'm wondering, what was the level of evidence, the strength of the evidence showing that it was superior as well? And are we confident, in that level of evidence that to prefer it over all other drugs for ulcerative colitis? Was it only, and I would have, I don't recall what those slides actually said from earlier. And was that a meta-analysis, a network analysis compared to all other drugs, or was it to one particular drug?
So this was a fair methodologic quality RCT comparing vedolizumab to adalimumab, and its findings were at 52 weeks. It had a study size of 769. So it's just one randomized control trial that looked at 4 different outcome measures.
And this is Donna, understanding the complexity of this particular class with all of these moving parts and different indications. One, we don't normally do this, but we could abstain from making a motion today and bring this back at the next meeting with where we have gone through the evidence and looked at the different indications, and we can put together maybe a table that shows, you know, all of the drugs, all of their indications, which would assist you in kind of untangling how to make what preferred.
This is Nancy. I would be okay with that. I would be in favor of that.
Yeah, this is Connie Nguyen. I'd be in favor of that as well.
This is Alex Park. Could I offer a counterpoint here? I mean, the head-to-head trials are usually one drug against another, so I feel uncomfortable making a recommendation that a particular drug is going to be preferred in that entire class when the data is primarily one drug against another.
This is Virginia. Sorry, Nancy. I think I'd like to this is Virginia again. I'd like to come back to Alex's point and just think about this from the perspective of giving the HCA the most flexibility and giving patients the greatest access to what they need. So I'm in support of wording something that keeps access open to patients for whatever they need. Maybe not whatever, but you know what I'm saying.
I mean, everything's always covered, so all of these drugs are covered. It's just a matter of, you know, is it covered? You know, do they need to try another drug before they would have access to the second drug? And you know, for Medicaid and for the Uniform Medical Plan, there's, you know, the costs are quite different, and which is why we now have different PDLs. So it may be. It would make it easier for us with more flexibility, because Medicaid does follow your motions, but when we do the cost analysis, we might end up with selecting different drugs.
So again, this is Virginia. I would probably tend to, not probably, I would stand towards working with a broader statement and going ahead and moving towards a motion today. However, I'm absolutely open to hearing if people don't feel comfortable with that and would like to take the option that Don has offered of waiting until our next meeting.
This is Susan. I agree. I feel like we should proceed with the motion today.
I agree. This is Alex. I agree.
This is Catherine. I agree also.
This is Alex. I think the list of FDA indications probably gets to some of your concerns there, Nancy, I hope, and also that of the committee, in that it's a fairly broad list of indications for which we are recommending that the PDL include a TIM for all of those. I don't see a diagnosis that I would want to make sure that we include so that patients have an access to a preferred TIM. I mean, of course, there are other diagnoses that could go on there. Kawasaki's disease is treated by anakinra and so forth and so on, but those are fairly unique cases that I don't feel we need to concern ourselves with in this motion.
This is Nancy. I appreciate everybody's input, and yeah, I would be okay with moving forward, just kind of hearing everything and all the different points being made, unless others want to chime in.
This is Alex Park. I just have one last question before I would feel comfortable making a motion. The difference between saying must include versus should include, what does that mean for the HCA?
This is Donna. We interpret that as to be the same. If you say should or must, we interpret it as must.
Okay. I only ask because it says must include a drug in terms of the FDA indications, but then it says should include in terms of the other concern that I think Susan had raised about self-administered. So those two would be considered equally powerful, so to speak, in terms of our motion. Okay.
Yeah, and we could change should to must.
I'd be okay with that. Okay, this is Alex Park. After considering the evidence of safety, efficacy, effectiveness, and special populations for the use of targeted immune modulators for their FDA-approved indications, I move that baricitinib, guselkumab, risankizumab, tildrakizumab, upadacitinib, abatacept, adalimumab, anakinra, apremilast, brodalumab, certolizumab, pegol, etanercept, godelimumab, infliximab, itolizumab, natalizumab, rituximab, saculimumab, secunizumab, tocilizumab, tofacitinib, ustekinumab, and vedolizumab are efficacious. The PDL must include a drug approved for the treatment of the following FDA indications: rheumatoid arthritis, juvenile idiopathic arthritis, ankylosing spondylitis, psoriatic arthritis, Crohn's disease, ulcerative colitis, and plaque psoriasis, and must include a self-administered agent if indicated. These medications cannot be subject to therapeutic interchange in the Washington preferred drug list.
This is Jordan Storhaug. I second.
This is Virginia Bucola, committee chair. All in favor?
Aye. Aye.
And the motion carries. Thanks to the committee for that discussion, and thanks to Alex for tackling all of those drug names. So we're scheduled to go to lunch. We were going to do a 30-minute lunch, and so it's 11:56. Shall we meet back here at 12:30 to round up?
That sounds good. Jenny, before we go, this is Lita. There were two questions about the sickle cell anemia being canceled for today. As far as being informed as to when it will be discussed, you can sign up on our website on the stakeholder list so that you will be emailed the agenda 30 days before the meeting. Donna, do you know if that will be reviewed in the next meeting or is that on hold?
Hey, Lita, this is Donna. I think we're anticipating the December meeting. Ryan, is that sound accurate?
Hi, this is Ryan. Yes, I believe we're getting a DERP report on sickle cell anemia due in December, and we are planning on having sickle cell drugs be reviewed around that time. We can defer to Marissa since she's now planning the DUR reviews.
Yeah, so for now, I think December is what we will be targeting towards, and if that changes, just watch for the December agenda, and it should come out in early November.
All right, Emma, I think I'll go ahead and adjourn the meeting as long as there are no other questions, and we'll meet back at 12:30.
Sounds good. And I'm just going to pause the meeting so it will still be on so people don't have to log back in.
Thank you.
All right. See you at 12:30. And everyone, please mute your microphones.
Hey, Lita, this is Donna. Do we want to put up a slide that says we're on break for lunch and that we'll resume at 12:30?
Yes.
Okay, great. Thank you.
Hi, Diane.
Hello, everybody. We're coming back from lunch and going to go ahead and adjourn the DUR or convene. We're not going to adjourn. We already did that. We're going to convene the DUR board. And I know I'm jumping right in, so I don't know, Umang, if you feel ready to go yet. So whenever you're ready.
Absolutely. Can you hear me okay?
Yes.
Okay, great. All right. So just to give a little bit of a refresher, the therapeutic classes that we will discuss, if you go to the next slide, there will be an overview of disease state, indications, dosage forms, and guideline updates. We have kind of reformatted this a little bit based on committee feedback, and it will be different from the past. We will only be reviewing new information, such as guideline updates, new drugs, additionally approved indications within the last 12 years. The first topic we will, on the next slide, will be hematological oral oncology medications. On the next slide here, again, to give a bit of background, there is a lot of information here, and one of the feedbacks I did receive was to kind of summarize some of these if possible. So what I will do is I won't, read through this one by one, but I'll kind of just I'll go over the main pearls, if you will, for some of these slides. So as one can imagine, there are, for hematological oral oncology, there are 7 sub-disease states that fall into this that will be presented. The first 3 on this slide are AML, CML, and multiple myeloma. So for AML, which is acute myeloid leukemia, the most common form of acute leukemia among adults estimates roughly less than 6,000 cases diagnosed and 1,500 deaths in the US in 2019. In patients who obtain a CR, 3-year survival rate is 45%. Remission rates are inversely proportional to age. For the next disease state for CML, for chronic myeloid leukemia, it comprises of 15% of all adult leukemia. There are 3 phases of this disease, which are chronic, accelerated, and blast. The gene mutation called the Philadelphia chromosome has been identified, which involves a translocation T from the 9 to the 22 chromosome, also known as the BCR-ABL translocation. And the discovery of tyrosine kinases that inhibited the BCR-ABL has revolutionized the treatment of CML, making long-term remission a reality. And the final disease state here, we have multiple myeloma, which accounts for about 15% of all hematologic malignancies. It usually responds to initial chemotherapy, but responses are often transient. On the next slide here, we have the next 3 being NHL, non-Hodgkin's lymphoma. This is a lymphoma. Lymphomas are heterogeneous groups of malignancies that originate in the immune cells, predominantly beta cells and T cells, of the lymphoid tissue. Leukemias and lymphomas are similar diseases with overlapping characteristics. Most lymphomas involve tumor invasion of the lymph nodes and other tissues, while the malignant clone in most leukemias predominate in the bone marrow. And the most common presentation is that of a solid tumor. For the next one, we have chronic lymphocytic leukemia, CLL, and small lymphocytic lymphoma, which is SLL. And this is the most prevalent adult leukemia with a median age of diagnosis at 72 years of age. Treatment is individualized, as some patients may have indolent disease while others require treatment. And cytogenetic abnormalities are of prognostic significance and can help to drive the treatment options. For mantle cell lymphoma, or MCL, it possesses characteristics of both indolent and aggressive NHL, and the median onset is 3 years, but no evidence of survival plateau, and is similar to indolent leukemia. The 7th and the final, quote, disease state we will discuss is graft versus host disease on the next slide here. Although the exact cause is unknown, graft versus host disease is an autoimmune mediated disease that can result following complications of hematopoietic cell transplantation, in which the transplanted cells, which are grafts, recognize the recipient's body as foreign. Chronic graft versus host disease is generally an extension of acute graft versus host disease that often develops more than 100 days after transplant. Pivoting over to the guidelines for graft versus host disease, the American Society for Bone Marrow Transplantation has a clinical practice guideline around the first and second line treatment of acute graft versus host disease. They state that corticosteroids are the standard of care for the initial treatment of acute graft versus host disease, and note that literature does not support the choice of any specific agent for secondary therapy of acute graft versus host disease. These guidelines were published prior to May 2019, FDA approval of Jakafi for the treatment of corticosteroid refractory acute graft versus host disease in adult and pediatric patients aged 12 years of age and older. Moving onward to the next slide here, continuing on with guidelines for AML. Keep in mind that the guidelines for this are short. As one can imagine, there are a huge body of guidelines, but as I mentioned earlier, we're only reviewing the ones in the last year. So for AML, the NCCN guidelines in 2020 stated for standard induction to include Rydapt in combination with cytarabine, with or without daunorubicin, as part of standard induction, consolidation, and post-remission therapy for patients with FLT3 mutated AML. For intensive remission induction therapy, adult patients who are candidates for intensive remission induction therapy with unfavorable risk cytogenetics may receive Venclexta in combination with hypomethylating agents, decitabine and azacitidine, or cytarabine, which are all category 2A, while patients 60 years of age or older who are not candidates for intensive remission induction or who decline intensive remission and are without actionable mutations may be treated with venetoclax or and Daurismo, among other options. Venetoclax is also listed as an option with Idhifa or Tibsovo for IDH2 mutated or IDH1 mutated AML, respectively. Now, for post-induction therapy in patients who have a response to lower intensity therapy, the lower intensity regimen should be continued. And this includes the following medications listed. And lastly, for relapsed refractory AML, these patients who have IDH1 and IDH2 mutations, therapy with anasidinab and ivosidenib, respectively, may be utilized. And for patients with FLT3 ITD or FLT3 TKD mutations, Zospata is a category 1 recommendation. The guidelines note that these drugs increase the risk for differentiation syndrome and hyperleukocytosis, which may require treatment with hydroxyurea and corticosteroids to mitigate. Now, these guidelines note that in the event of relapsed or refractory disease after the completion of consolidation, targeted therapies may be retried if the drugs were not administered continuously and stopped due to the development of clinical resistance. On the next slide here, we do have, on the next slide, we have the subcategories based on Apple Health. And of these, you see there are 14 subclasses, first being the alkylating agents. We have antimetabolites, antineoplastic miscellaneous, BCL2 inhibitors. We have histone deacetylase inhibitors, immunomodulators, isocitrate dehydrogenase 1 inhibitors and 2 inhibitors. We have the JAK inhibitors, the PI3K inhibitors, proteasome inhibitors, the XPO1 inhibitors, and lastly, the immune modulators, specifically myelodysplastic syndromes. Now, if you notice, some of the drug names here are bolded, and the bold does indicate there is new information in the last year that we will be going over. So on the next slide, as I mentioned, there are no updates for the first 3 subclasses, but we'll move right along to Venclexta on the next slide here. Okay, so for Venclexta, in August 2019, the FDA alerted the public of a risk associated with the investigational use of Venclexta in patients with multiple myeloma, and it was an off-label use based on data from the Bellini clinical trial, which reported after a median follow-up of 17.9 months, an increased risk of death with venetoclax, roughly 21% compared to placebo, which is 11%, when used in combination with bortezomib and dexamethasone in patients with multiple myeloma. Now, again, just for the committee, I do want to remind, there's a lot of information here. Please note that the bolding will indicate things I am going to go over, but the other information is here just for completeness sake. Please know that the indications, the warning and precautions are still here. The dosing, if you would like to see the full prescribing information, it is in the TCR that is in the web portal, the Therapeutic Class Review, and the availability as well. To give a little background, venetoclax is a small molecule inhibitor with beta cell lymphoma 2, which is an anti-apoptic protein, and it restores apoptosis by binding to this protein, which is a part of the lymphadocytic leukemia cells. Please note there's no pediatric safety and efficacy established here, and there are warnings with tumor lysis syndrome, neutropenia, and infections, similar to a lot of the other medications in this class. Continuing on to the next slide, just continuing through the subclasses for Apple Health, there were no updates for the histone deacetylase inhibitors, excuse me, and the immunomodulators. On the next slide here, the next subclass we'll review are the JAK, the Janus-associated kinase inhibitors. And going to the next slide here, the first medication we will discuss is Inrebic. And so in August 2019, FDA approved Inrebic, a kinase inhibitor, for the treatment of adults with intermediate to or high-risk primary or secondary post-polycythemia vera or post-essential thrombocytopenia myelofibrosis. In terms of warning and precautions, a lot of these have similar black box warnings. This one here does have serious or fatal encephalopathy, including Wernicke's, which has occurred in patients treated with this medication, anemia and thrombocytopenia, as one can imagine, GI toxicity, and so this is managed by a dose reduction, interruption, or transfusion. Similarly, GI toxicity, hepatic toxicity was mentioned earlier with encephalopathy, amylase and lipase elevation, and as one can imagine, you would avoid this in patients who are compromised in said systems, such as hepatic impairment. Dosing and availability is here for your leisure. And to go over the mechanism of action, this is a kinase inhibitor with activity against a wild type and mutationally activated Janus-associated kinase 2, or JAK2. And essentially, this is a selective inhibitor with high affinity activity, and it helps reduce, without going into kind of the biochemistry of it, it helps, it inhibits cell proliferation as seen in cancer, and it induces apoptosis. The next slide here, we do have another JAK inhibitor, it's Jakafi, and in May 2019, FDA approved an expanded indication for steroid refractory acute graft versus host disease, which I mentioned earlier in the background, in adult and pediatric patients 12 years of age and older. As you can see, it had other indications as well, such as myelofibrosis, polycythemia. The warnings and precautions, I won't continue going over them because I'll sound like a broken record, but I will try to highlight the ones that may be different from the ones you have heard. There is a risk of non-melanoma skin cancer, so it is recommended that healthcare practitioners perform periodic skin examinations, and it can elevate lipid levels. All right, continuing onward. Looking at the Apple Health subclasses here, there are no updates for the PI3K inhibitors or the proteasome inhibitors, so we'll move right along to the XPO1 inhibitors, and we have a medication called Xpovio. Now, in July 2019, FDA approved this medication, which is a nuclear export inhibitor indicated in combination with dexamethasone to treat adults with relapsed or refractory multiple myeloma who have received 4 or more prior therapies and whose disease is refractory to 2 or more proteasome inhibitors, 2 or more immunomodulator agents, and an anti-CD38 monoclonal antibody. Very similar warnings and precautions of thrombocytopenia, neutropenia, and GI adverse effects, and dosing and availability is found in the TCR. To take a step back for the mechanism of action, this medication through a pathway causes apoptosis of cancer cells by inhibiting the tumor suppressor protein. So it inhibits the suppressor, and it inhibits growth regulators along with oncogenic proteins as well. Continuing on, the last updated medication class we'll talk about is the myelodysplastic syndromes for the immunomodulators, which would be Revlimid. And on the next slide here, you do see for Revlimid in October 2019, FDA approved Revlimid in combination with a rituximab product for previously treated follicular lymphoma and previously treated marginal zone lymphoma. As you can see, it has a litany of other indications. Or black box warning, it does have an embryo fetal toxicity, hematologic toxicity, specifically with neutropenia and thrombocytopenia, and venous and arterial thromboembolism. Now, this medication is an analog of thalidomide, and it is a category X, and so that is why it does carry that embryo-fetal toxicity, and pregnancy must be excluded in patients at the start of treatment, and 2 reliable forms of contraception must be used in patients who are of childbearing age. That concludes the hematologic oral oncology medications. Any questions there?
This is Virginia, just double checking, no questions. Thanks, Umang. We just have one stakeholder. It's Dr. Margaret Ullman with AbbVie. If there are any other stakeholders, please go ahead and raise your hands. And we'll go ahead and go to Dr. Ullman, and you have 3 minutes for your comments.
This is Lita. I don't see Margaret Ullman still on.
Okay.
Oh, wait, I'm sorry. Here she is. Okay, sorry, she got moved to the bottom. Sorry, Margaret. Okay.
No problem. No problem at all. I was trying to wave a hand or something to see if you could see me. I want to thank Dr. Patel for such a good summary of so many very difficult topics to look at all at once here. Again, my name is Dr. Margaret Ullman with U.S. Medical Affairs at AbbVie, and I want to thank you for the opportunity to provide a short summary for information about venetoclax, brand name Venclexta. Again, it's a short summary, so please see the prescribing information at rxabbvie.com for complete safety and efficacy details. A fixed duration of Venclexta treatment is indicated for patients with chronic lymphocytic leukemia and small lymphocytic leukemia. Venclexta is also FDA approved in combination with azacitidine or decitabine or low-dose cytarabine for the treatment of newly diagnosed acute myeloid leukemia in adults 75 years or older, or in patients who have comorbidities that preclude use of intensive induction chemotherapy. Venclexta is a selective and orally bioavailable small molecule inhibitor of BCL2. Overexpression of BCL2 has been associated with resistance to chemotherapy agents. Venclexta helps restore the process of programmed cell death. It's been shown to improve patient response when used either as monotherapy or in combination with other oncology treatments. In 2 phase 3 studies in CLL, patients treated with combinations that included a fixed duration of Venclexta treatment showed a significant improvement in progression-free survival. In 2 phase 3 clinical trials in AML, in patients on combinations that included Venclexta, the complete response rates ranged from 21 to 54%, and time to full or partial response for between 1 or 2 months. Safety of these Venclexta combinations is consistent with what has been seen in previous clinical trials. Patients taking Venclexta should be assessed for tumor lysis syndrome risk, including evaluation of tumor burden and comorbidities, and should receive appropriate prophylaxis measures, including hydration, anti-hyperuricemics, and laboratory monitoring. Venclexta is the only oral chemo-free regimen that offers sustained treatment-free remission across all lines of CLL therapy and is a preferred regimen for first-line and relapsed refractory therapy in the NCCN guidelines. NCCN also recommends the 3 Venclexta combination therapies as first-line treatments in patients with newly diagnosed AML, in patients who are 60 years or older who have unfavorable risk cytogenics or who are unable to take intensive chemotherapy. In summary, I'm requesting the committee continue to have Venclexta available for oncology patients in Washington. Thank you for your time and your consideration. Be happy to answer any questions.
Thanks, Dr. Ullman. Any questions from the committee? Okay, it looks like we can go ahead and move to the motion.
This is Lita. Marissa, do you want to walk them through the motion?
Sure. So I just lumped everything for the oncology agents for the different subclasses onto one slide. So it'll be one motion for all of them. So if you want to go to the next slide, Lita. And all the drug classes are listed on slide 2. So if you want to see those drug classes again, just let me know. Any questions?
This is Susan. I move that all products in the drug classes listed on slide 2 are considered safe and efficacious for their medically accepted indications and are eligible for preferred status and grandfathering at the discretion of HCA. Products in this class may require prior authorization to determine medical necessity. All non-preferred products require a trial of 2 preferred products with the same indication before a non-preferred drug will be authorized unless contraindicated, not clinically appropriate, or only one product is preferred.
I second that. This is Nancy Lee. I second that motion.
This is Virginia, committee chair. All those in favor?
Aye.
Aye.
And the motion carries. And we're going to move next back to Umang to discuss cytokine and CAM antagonists.
Okay, great. All right, so for the cytokine and CAM antagonists, I know the committee in the morning half of this meeting saw that there is an abundance of disease states that kind of fall into this umbrella of cytokine and CAM antagonists. I did my best to kind of do the summary. However, it wasn't always feasible because there is a lot of information here, especially since there's so many disease states. So for the first one, on the next slide here, we'll just do a quick background. So cytokine and CAM antagonists are chemical mediators involved in inflammatory processes throughout the body. Cytokines are small proteins secreted in response to an immune stimulus for the purpose of mediating and regulating immunity, inflammation, and hematopoiesis. The actions of the individual cytokines are widely varied, and they contribute to fibrosis and tissue degeneration associated with chronic inflammation, primarily by inducing proliferation of fibroblasts and collagenase. The pro-inflammatory cytokines, tumor necrosis factors, or TNF, and interleukin-1, or IL-1, are involved in tissue destruction in many chronic inflammatory diseases affecting various organs. On the next slide here, CAMs, or cell adhesion molecules, are cell surface proteins involved in the binding of cells, usually leukocytes, to each other, endothelial cells, or the extracellular matrix. Specific signals produced in response to wounds and infection control the expression and activation of these molecules. CAMs fall into 3 general family of proteins. We have the immunoglobulin superfamily, the integrin family, and the selectin family. Now keep in mind, different CAMs have been implicated in inflammatory, fibrotic, and autoimmune diseases, respective of said disease. On the next slide, the first disease we'll look at is ulcerative colitis. It is a chronic inflammatory disease primarily affecting the colon and rectum. It affects approximately 1 million people in the United States, and the incidence continues to increase worldwide. The CDC estimates the current prevalence of ulcerative colitis is 238 per 100,000 adults. Now, the predominant symptoms of ulcerative colitis is diarrhea, which is usually associated with blood in the stool. Additional symptoms may include pain in the lower quadrant or rectum, along with systemic features like a fever, malaise, and weight loss. The initial attack of ulcerative colitis may be fulminant with bloody diarrhea, but the disease more commonly begins indolently with non-bloody diarrhea progressing to bloody diarrhea. Most commonly, it follows a chronic intermittent course with long periods of quiescence interspersed with acute attacks lasting weeks to months. The primary goal of treating ulcerative colitis is essentially inducing then maintaining remission of the disease. On the next slide, normally I do background and then guidelines, but like I said, there's so many disease states, I figured For the committee's sanity on trying to stick with a topic, I'm going to do background and guidelines if it's relevant in the last year. So American Gastroenterological Association, or AGA, in 2019, they stated that, again, agents in this class are not addressing the recommendation for induction and maintenance of mildly active disease. For moderate or severe active ulcerative colitis, specifically induction of remission, the group recommends oral systemic corticosteroids. TNF antagonists are also recommended, and if infliximab is used, it should be used with thiopurine. Notably, these guidelines state that robust data of combining TNF antagonists and immunomodulator therapy in moderately to severely acute ulcerative colitis exists only for infliximab and thiopurines. Vedolizumab or tofacitinib is recommended in patients who have previously failed TNF antagonist therapy. And in patients who were previously TNF antagonist responders but are subsequently having inadequate response, they recommend monitoring the serum drug level. In addition, the group states that patients who are primary non-responders to TNF antagonists should be considered for an alternative mechanism of disease control rather than a switch to another TNF antagonist. However, for secondary failure, initial response to TNF antagonists with later loss of efficacy, another TNF antagonist may be used. And to maintain remission in patients with previously moderate to severely active ulcerative colitis, these guidelines recommend the addition against the addition of 5-ASA in patients on TNF antagonists in those who had previously failed 5-ASA, continuing adalimumab, golimumab, or infliximab if used to achieve remission, continuing vedolizumab if used to achieve remission, and continuing tofacitinib if used to achieve remission. Several other recommendations were detailed in the guidelines, including the role of medications not within this class or non-pharmacological guidance. Pivoting over to juvenile idiopathic arthritis on the next slide here, we have the organization recommends NSAIDs conditionally as adjunctive therapy. Regarding traditional DMARDs for polyarthritis, methotrexate is conditionally recommended over leflunomide or sulfasalazine, and subcutaneous methotrexate is conditionally recommended over oral methotrexate. Combination therapy with a DMARD is conditionally recommended over biologic monotherapy when initiating treatment with a biologic. Combination therapy with a DMARD is strongly recommended for infliximab. Intra-articular glucocorticoids are conditionally recommended as adjunct therapy, and oral corticosteroids as a bridge therapy are conditionally recommended in patients with moderate or high disease activity. However, bridge therapy is not recommended in patients with low disease activity. And in addition, the group strongly recommends against adding chronic low-dose glucocorticoids regardless of disease activity. Continuing on to the next slide with these guidelines, for initial therapy with polyarthritis patients, they state that all patients have initial therapy with DMARD over NSAID monotherapy. In patients without risk factors, which are positive anticyclic citrulline peptide antibodies, positive rheumatoid factor, or presence of joint damage, they recommend initial therapy with a DMARD conditionally over a biologic. However, in those with risk factors, the group recognizes that there are situations in which a biologic may be preferred. For subsequent therapy in low disease activity patients, defined as clinical juvenile disease activity score based on 10 joints, they do recommend escalation of therapy. For example, intra-articular glucocorticoid injection, DMARD, dose optimization, methotrexate trial, and adding a biologic is recommended over no escalation. For subsequent therapy in moderate or high disease activity, and that's based on that SED score I mentioned a second ago, patients receiving DMARD monotherapy, it is recommended adding a biologic to the original DMARD over changing to a second DMARD or triple DMARD therapy. For subsequent therapy in moderate or high disease activity polyarthritis patients receiving a TNF antagonist with or without a DMARD, the group recommends switching to a non-TNF antagonist such as tocilizumab or abatacept over switching to a second TNF antagonist. However, the group recognizes that a second TNF antagonist may be appropriate in patients with good initial response to TNF antagonists who have experienced secondary failure. And lastly, if the patient is receiving their second biologic, use of TNF antagonists, abatacept or tocilizumab is conditionally recommended over rituximab. The next slide here, again, continuing with this said guideline for patients with juvenile idiopathic arthritis or sacroiliitis, guidelines strongly recommend treatment with NSAID over no NSAID treatment. In those already on NSAIDs with continued active disease, the group strongly recommends a TNF antagonist over NSAID monotherapy. They continue to recommend against the use of methotrexate monotherapy. Bridging therapy with a limited duration oral corticosteroid in select conditions and adjunct use of intra-articular glucocorticoid are conditionally recommended. Those with JIA and enthesitis, the group recommends NSAID treatment over no NSAID treatment with a TNF antagonist conditionally recommended over methotrexate or sulfasalazine. Bridging therapy with a limited duration oral corticosteroid in select conditions. And the group provides additional recommendations on SEDS glucocorticoids as well. Now, moving on to the next slide, we have pediatric psoriasis. And in 2019, the American Academy of Dermatology and National Psoriasis Foundation published guidelines for the management of psoriasis in pediatrics. They recommend ongoing assessment for psoriatic arthritis, uveitis, obesity, CV risk factors, dyslipidemia, insulin resistance, diabetes, and mental health conditions. Body surface area plus children's dermatology life quality index should be used to assess the disease severity. And in terms of treatment, they recommend topical treatments for psoriasis, and those include topical corticosteroids, which is off-label, tacrolimus 0.1% ointment for face and genital regions. We have calcipotriene, calcipotriol, calcipotriol beta-methasone combination, tazarotene plus topical corticosteroids, topical anthralin, coal tar, phototherapy, and photochemotherapy. They do recommend systemic treatment, which could be methotrexate, cyclosporin, systemic retinoids, and biologics such as etanercept, infliximab, adalimumab, and ustekinumab. And lastly, the guidelines recommend treatment of physical and psychosocial wellness, such as quality of life, for these pediatric patients as well. Moving on to the next subdisease class, we have ankylosing spondylitis. And this is an inflammatory condition generally affecting the spine and can be further subdivided into ankylosing spondylitis and non-radiographic ankylosing spondylitis represented here. Now, the American College of Rheumatology, Spondylitis Association of America, and Spondyloarthritis Research and Treatment Network created a joint guideline update in 2019 on the treatment of both ankylosing spondylitis and non-radiographic axial spondyloarthritis. In general, they recommend recommendations for AS and non-radiographic axial are similar. TNF antagonists, but not a specific one, are recommended as first biologic over Cosentyx or Tremfya, which are then recommended over a second TNF antagonist if the first one does not produce a response. All the prior mentioned agents are recommended over Xeljanz. Concurrent low-dose methotrexate with TNF antagonist is not recommended. They do recommend against a strict treat-to-target strategy. If a patient's disease is stable, guidelines recommend against discontinuing or tapering of biologics. And sulfasalazine provides a viable option for select patients who cannot take a TNF antagonist. Continuing on to the next disease state, we have periodic fever syndrome. And these are rare hereditary syndromes that are characterized by short and recurrent severe localized inflammation and fever, quote, attacks that are not otherwise explained by routine childhood or even adult infections. It is defined as 3 or more episodes of unexplained fever within a 6-month period, occurring at least 7 days apart. And these could be periodic or they could be irregular. Cryptopyrene-associated periodic syndromes, or CAPS, is a family of syndromes associated with mutations in this cryptopyrene, now known as nucleotide binding domain and leucine-rich repeat-containing family, pyrene domain containing 3, or NLRP2. And these include things such as Muckle-Wells syndrome, familial cold autoinflammatory syndrome, and chronic infantile neurologic cutaneous articular syndrome, which is also known as neonatal onset multisystem inflammatory disease. So the treatment for this, well, Kineret, Ilaris, and Arcolast are approved for the treatment of CAPS in select age. Kineret is only approved for patients with CAPS associated with NOMID. and Arcalyst and Ilaris are approved more generally for patients with CAPS, including FCAS and MWS, the sub-diseases I mentioned earlier. Ilaris is also approved for the following other periodic fever syndromes, such as tumor necrosis factor receptor associated periodic syndrome, hyperimmunoglobulin D syndrome, and mevalonate kinase deficiency, along with familial Mediterranean fever. On the next slide here, the next disease state we have is giant cell arteritis, or also known as temporal arteritis. It is a systemic inflammatory vasculitis of unknown etiology that is classified as a large vessel vasculitis, but typically also involves small and medium arteries. Most commonly, it affects the occipital and ophthalmic, posterior ciliary, proximal, vertebral, and vertebral arteries. While the incidence of this ranges from 0.5 to 27 cases per 100,000 people in those 50 years of age or older, the incidence is higher in the northern areas of the U.S. And it occurs in older persons and can result in a wide variety of neurologic, ophthalmologic, and systemic complications. The treatment is just high-dose corticosteroids, although clinical studies on various dosing protocols are limited. Steroid are generally continued until the resolution of symptoms, and then may be tapered slowly to the lowest dose that adequately suppresses symptoms. And Actemra is the only non-corticosteroid drug FDA approved for the treatment of giant cell arteritis. On the next slide here, we have hidradenitis suppurativa, and this is a chronic condition that affects the terminal follicular epithelium in the apocrine gland-bearing skin, such as armpits and perianal area. It occurs in adolescents, generally after puberty, and adults, and it's about 1 to 2% of the population. Signs and symptoms are things such as erythema, raised bumps or lesions, painful lesions, and a local arthritis or arthralgia. Now, the European Dermatology Forum in 2015 stated that guidelines for this are limited, but guidelines recommend either adalimumab or infliximab in severe or refractory disease, stating adalimumab seemed to be much more tolerated. However, only adalimumab is the FDA-approved medication for this use. Continuing with uveitis, it is a non-infectious intermediate and posterior uveitis, inflammation of the intermediate and posterior uvea, while panuveitis is the inflammation of the anterior chamber, vitreous humor, and choroid or retina simultaneously. Together, these represent the most severe and highly recurrent forms of uveitis. The incidence of all cases of uveitis is approximately 15 cases per 100,000 patients per year, and anterior uveitis is the most common form of uveitis. Continuing on to the guidelines, in the last year, ACR and Arthritis Foundation released this guideline, and they recommend topical glucocorticoids in patients with JIA and active chronic anterior uveitis for short-term control. But for those unable to control the symptoms, they recommend adding systemic therapy in order to taper topical glucocorticoids. Regarding specific agents, they recommend subcutaneous methotrexate conditionally over oral methotrexate. However, the use of a TNF antagonist with methotrexate in severe active disease and sight-threatening complications is conditionally recommended. If starting a TNF antagonist, they conditionally recommend a monoclonal antibody over etanercept, abatacept or tocilizumab as biologics, and mycophenolate, leflunomide, cyclosporin as non-biologic options are conditionally recommended in patients who have failed methotrexate and 2 monoclonal antibody TNF antagonists. The disease should be well controlled for 2 years on a DMARD before considering tapering. For pediatric patients who develop acute anterior uveitis, the group conditionally recommends topical glucocorticoids prior to change in systemic therapy. And notably, the only agent approved for uveitis in this class is adalimumab. Continuing on to our last background guideline slide, we have cytokine release syndrome, which is a condition that can occur following select immunotherapies and can result in a large, rapid release of cytokines into the blood. This can manifest as fever, nausea, headache, rash, tachycardia, hypotension, and dyspnea, and as you can imagine, can be life-threatening. Actemra is approved for the treatment of CAR T cell-induced severe or life-threatening CRS in adult and pediatric patients 2 years of age and older. And lastly, for the role of biosimilars, Although this ACR document is over a year old, I felt it important to keep it in here as questions sometimes do arise about biosimilars. The ACR published a white paper regarding the use of biosimilars in the treatment of rheumatic disease, and they provided a comprehensive overview. They note that available real-world studies have demonstrated efficacy and extrapolated indications and state that healthcare providers should incorporate biosimilars where appropriate into treatment for patients with rheumatologic diseases. They state treatment is a shared decision between patient and clinician, and patient and providers must be educated on the biosimilars. And in addition, biosimilars are not considered superior or inferior to the originator product, and biosimilars should be considered safe and effective for all the originator products' approved indications. However, they do caution against interchangeability without consultation with a prescriber. Now, before we move on to the next slide, which has our first new updated indication for medication, I do want to let the committee know that a lot of the other guidelines that we did not review because they were over a year old are found in the appendices. I did add it there for the committee's leisure if they wanted to go through that, and that is located at the end of this slide deck. Okay, so moving onward to the next slide. First, we have Cimzia here. So in March 2019, FDA approved the expanded indication of Cimzia for the treatment of adults with non-radiographic axial spondyloarthritis with objective signs of inflammation for the treatment of adults with active ankylosing spondylitis. Keep in mind the limitations for a lot of these cytokine and CAM antagonists will be very similar. There are things such as live vaccines should not be given with this medication. There are black box warnings such as serious infections and malignancies. So if a patient is at an increased risk of serious infections leading to hospitalization or death, including TB, bacterial sepsis, invasive fungal infections such as histoplasmosis, and infections due to other opportunistic pathogens. In terms of malignancies, lymphoma, and other malignancies, some fatal, have been reported in children and adolescent patients treated with TNF blockers. And in terms of tuberculosis, it is important to evaluate the patient for TB prior to initiating treatment. This medication's dosing is stratified by indication and weight, and that is found in the package insert, or for the committee, it is found in the TCR in the web portal that you have. To give a little bit of background, the mechanism of action of this medication, so TNF-alpha is a key pro-inflammatory cytokine that plays a role in inflammatory process. This medication selectively neutralizes that said protein, and it cross-reacts it inhibits the TNF-alpha protein. In terms of pediatrics, the safety and efficacy of this medication has not been established in patients less than 18 years of age. And in terms of patients who have renal or hepatic impairment, specific clinical studies have not been performed to assess the effect on renal and hepatic impaired patients. All right, and the next one we have here is Skyrizi on the next slide. So for Skyrizi, we have in April 2019, FDA approved this medication for the moderate to severe treatment of plaque psoriasis in adults who are candidates for systemic therapy or phototherapy. The limitations, again, live vaccines should not be given with this medication. Additional specific contraindications per their PI has not been determined. The dosing and availability is here for you. I'll give similar information I gave for the previous medication. In terms of pediatrics, safety and efficacy has not been established, and no specific studies have been established for hepatic or renal impaired patients. And in terms of the mechanism of action, this medication is a humanized immunoglobulin, and it binds to a specific P19 subunit, which then in turn goes to the IL-23. Essentially, it helps mediate that inflammatory pathway process that you see. On the next slide here, we have Inflectra. So in June 2019, FDA expanded the indication of Inflectra to also include reducing signs and symptoms and inducing and maintaining clinical remission in pediatric patients 6 years of age or older with moderately to severely active ulcerative colitis who have an inadequate response to conventional therapy. It already had other indications, as you can see. So again, the limitations are very similar. We have increased risk of infection, lymphoma, and other malignancies. This one does have a post-marketing case of fatal hepatosplenic T-cell lymphoma. and similar to others of serious infection. The dosing again is stratified by indication and weight. Specific clinical studies have not been performed for hepatic and renal impaired patients. Moving onward to the next medication in this class that we'll review is Renflexis. In June 2019, the FDA approved expanded indication to reduce the sign and symptoms of inducing and maintaining clinical remission in pediatric patients 6 years of age or older with moderately to severe active ulcerative colitis who have had an inadequate response to conventional therapy as well. Again, similar to the prior medication, there are a litany of other indications for this medicine. Almost identical limitations that I won't go over, and dosing again is stratified by indication and found in the TCR. There is no available data for this medication and its effect on women who are pregnant. For patients who have severe hepatic reactions, including acute liver failure, jaundice, hepatitis, and colitis, that has been reported in post-marketing data. However, no specific studies have been conducted to determine the effect on renal or hepatic impaired patients prior to this medication. On the next slide here, we have Otezla. In July 2019, the FDA approved a new indication to treat adults with oral ulcers associated with Behçet's disease, and dosage for this indication is consistent with its other indications as well. In terms of limitations, there are some that are a little unique from the ones we've mentioned before: diarrhea, nausea, and vomiting. And if this does occur with this medication, consider dose reduction or suspension. Depression. Advise patients, their caregivers, and families to be alert for the emergence or worsening of depression, suicidal thoughts, or other mood changes. And weight decrease. So it is important that healthcare providers monitor weight regularly. If unexplained or clinically significant weight loss occurs, evaluate and consider discontinuation. Okay. Moving on to the next new medication here, we have Hadlima. In July 2019, Hadlima was approved, a biosimilar to Humira, for the treatment of rheumatoid arthritis, juvenile idiopathic arthritis, plaque psoriasis, psoriatic arthritis, ankylosing spondylitis, adult Crohn's disease, and ulcerative colitis. Please note that the launch is expected on or after June 30th of 2023. Now, the black box warnings are similar of Humira. So there are serious infections, malignancies, post-marketing cases of hepatosplenic T-cell lymphoma, and serious infections. Again, dosing stratified by indication and weight. In terms of renal impairment and hepatic impairment, no PK data available to show its effect on patients who had a baseline renal or hepatic impairment. Moving forward with Rinvoq. So in August 2019, FDA approved Rinvoq, a Janus kinase inhibitor. And this is for the treatment of adults with moderate to severe active rheumatoid arthritis who have had an inadequate response or intolerance to methotrexate. Now, the limitation of use, the use in combination with other JAK inhibitors, biologic DMARDs, or with potent immunosuppressants such as azathioprine and cyclosporin is not recommended. The limitations, again, are similar. Serious infections may occur. A lymphoma or other malignancies have been observed. A newer one is thrombosis, including deep vein thrombosis, pulmonary embolism, and arterial thrombosis have occurred in patients treated with Janus kinase inhibitors used to treat inflammatory conditions. And similar to the other medications, a TB test is required prior to the start of this treatment. And the dosing and availability can be found here as well. Since I've already mentioned the mechanism of JAK before, I'm not going to go into that. In terms of pediatrics, there is no study to show the safety and efficacy of this medication in patients less than 18 years of age. No dosage adjustment is required. No dose adjustment is required in patients with mild, moderate, or severe renal impairment, or in patients with mild or moderate hepatic impairment. However, it is not recommended in patients with severe hepatic impairment. Continuing to the next one, we have Olumiant. So in October 2019, FDA approved a new formulation of Olumiant 1 milligram tablet for dose adjustment in patients with moderate renal impairment or patients taking strong OAT3 inhibitors. So the indication for this is treatment of adult patients with moderate to severe rheumatoid arthritis who have had an inadequate response to one or more TNF antagonist therapy. So the dosing normally is 2 milligrams. However, this new expanded formulation came out of 1 milligram tablet, which is used, like I mentioned earlier, to patients who have moderate renal impairment, which is defined as creatinine clearance of 30 to 60 milliliters per minute, or patients who are taking strong OAT3 inhibitors. This medication has not been studied there's no safety and efficacy for patients who are less than 18 years of age. And as I mentioned earlier, there's a dose adjustment needed for renal impairment, no dose adjustment for mild or moderate hepatic impairment, but it is not recommended in patients with severe hepatic impairment. Continuing onward, we have Erelzi. And so in October 2019, the FDA approved a new indication for the treatment of psoriasis and psoriatic arthritis in adults. And it was already approved for rheumatoid arthritis, ankylosing spondylitis, and polyarticular juvenile idiopathic arthritis. Identical limitations and black box warnings. The dosing is bolded for the respective new indications it received. Pregnancy, the available studies do not reliably support an association between this medication and major birth defects. This medication has not been studied in patients less than 2 years of age, since, as you can see, the indication is for 2 years and older. And there are no formal PK studies for this medication in hepatic and renal impairment, so there are no recommendations there. Moving onward to Stelara. In October 2019, FDA approved a new indication for the treatment of moderate to severe active ulcerative colitis in adults. Again, it has a litany of other indications such as plaque psoriasis, psoriatic arthritis, and Crohn's disease. Identical limitations as the previous medications as well. There is a pregnancy registry that monitors pregnancy outcomes in women exposed to Stelara during pregnancy, and there is limited data on this medication in pregnant women to inform a drug-associated risk. In terms of pediatrics, it's got pediatric indications for certain disease states. So the safety and efficacy of Stelara has been established for pediatric patients 12 years of age to 17 years of age with moderate to severe plaque psoriasis, but it has not been established for any of its other indications. And no hepatic or renal impairment dose adjustments. Continuing to the next one, we have Abrelata. In November 2019, FDA approved this medication, which is a new biosimilar to Humira. It has identical indications: treatment of RA, juvenile idiopathic arthritis, psoriatic arthritis, ankylosing spondylitis, adult Crohn's, ulcerative colitis, and plaque psoriasis. Identical limitations as well with vaccines, the serious infections and malignancies. There is kind of a new limitation here that hasn't been mentioned before, an invasive fungal infection. So for patients with systemic illness on Abrelata, consider empiric antifungal therapy for those who reside or travel to regions where mycoses are endemic. Dosing is weight and indication based. And there is no formal recommendations for hepatic renal impairment. There is no reliable association between this medication and major birth defects as well. Alrighty, continuing, marching onward, we have Tremfya. In November 2019, this was FDA approved a new formulation as a single dose, one press patient-controlled injector, and it's a single dose prefilled syringe was already approved in the past. It is indicated for the treatment of adult patients with moderate to severe plaque psoriasis who are candidates for systemic or phototherapy. Again, identical limitations. and there's not too much information here, but the reason this was included into the slide set is because there is a new formulation. Anytime a new dosage formulation, black box warning, post-marketing study, anything is found, it will be included when this class is re-reviewed. Moving onward, so then we have in December 2019, FDA approved Avsola, which is a biosimilar to Remicade. And this, the indications for this include Crohn's disease in adult and pediatric patients, ulcerative colitis in adult and pediatric patients, rheumatoid arthritis in combination with methotrexate, ankylosing spondylitis, psoriatic arthritis, and plaque psoriasis. Identical limitations and black box warnings, as we've already mentioned before. Please note that hepatotoxicity has been seen with this medication, so severe hepatic reactions, some fatal or necessitating liver transplantation. And if a patient does appear to have jaundice and or marked liver enzyme elevation, the healthcare provider should stop this medication. Dosing is stratified by indication and weight-based, and it can be found in the TCR as well. Okay, moving onward to Xeljanz and Xeljanz XR. So in December 2019, the FDA issued a safety announcement regarding both Xeljanz and Xeljanz XR. An ongoing clinical trial found an increased risk of blood clots in the lungs and death when a 10 milligram twice daily dose was used in patients with RA, which is not an FDA-approved dose. Patients should be monitored for a pulmonary embolism and advised to seek medical attention if symptoms of an pulmonary embolism occur. At that same month, and to save a slide for the committee, that same month, the FDA also approved a new indication for Xeljanz XR for the treatment of adult patients with moderate to severe ulcerative colitis who have an inadequate response or who are intolerant to TNF blockers. There's no other extra information here, but this is for completeness sake for the committee. Moving onward to Xevi. In January 2020, FDA approved this medication for reducing signs and symptoms and inducing and maintaining clinical remission in pediatric patients 6 years of age or older with moderate to severe ulcerative colitis who have had an inadequate response to conventional therapy. And this was previously only indicated for adults with ulcerative colitis. It also does have other indications, as you can see, identical limitations, and the dosing is stratified by weight and indication that can be found in the TCR. All right, and our last slide for this therapeutic class is Taltz. In August 2019, the FDA approved the expanded indication of Taltz for the treatment of adults with active ankylosing spondylitis. And then roughly about a few months later, in March 2020, they approved Taltz for use in patients 6 years of age or older with moderate to severe plaque psoriasis who are candidates for systemic therapy or phototherapy. And previously, this was only found in adults. So as you can see, there are other indications found as well. Identical limitations. Please note in terms of one unique limitation here, Crohn's disease and ulcerative colitis, including exacerbations, did occur during clinical findings. So patients who are treated with Taltz and have inflammatory bowel disease should be monitored closely by the healthcare practitioner. Again, dosing is stratified by its indication, which is found in the TCR. And that concludes the cytokine and CAM antagonist updates. Any questions from the committee?
This is Virginia. Thanks, Umang. I know that was detailed. Yeah.
No problem.
Again, opening it back up to the committee for questions. Okay, if there are no questions, it looks like we have one stakeholder, and it is Dr. Margaret Ullman with AbbVie. So Dr. Ullman, if you're ready, you could go ahead and have your 3 minutes.
Thank you for the opportunity to speak with you once again. My name is Dr. Margaret Ullman with Medical Affairs at AbbVie. AbbVie now has 3 targeted immunomodulating medications available. I'd like to briefly review only the indications for Skyrizi, Rinvoq, and Humira and answer any questions you might have. Please see the full prescribing information online for comprehensive safety and efficacy data. Skyrizi is an IL-23 inhibitor indicated for the treatment of moderate to severe plaque psoriasis in adults and is given as a subcutaneous injection at week 0, 4, and every 12 weeks, which means maintenance treatment 4 times a year. Rinvoq is an oral JAK inhibitor indicated for the treatment of adults with moderately to severely active rheumatoid arthritis, and it's given as a 15 milligram tablet once daily. All TNFs, including Humira, carry similar boxed warnings regarding serious infections, tuberculosis, and malignancies. JAK inhibitors also include warnings about thrombosis. Patients starting any of these medications should be screened for TB and carefully monitored for serious events. Humira has 10 currently approved indications, including rheumatoid arthritis, juvenile idiopathic arthritis, psoriatic arthritis, ankylosing spondylitis, ulcerative colitis, adult and pediatric Crohn's disease, plaque psoriasis, uveitis, and hidradenitis suppurativa. In summary, I respectfully urge the committee to maintain a preferred status for Humira on the PDL and to add both Skyrizi and Rinvoq as available treatments for the Medicaid patients in Washington. May I answer any questions for you at this time? Thank you so much.
Thank you. We'll go back to Umang, and we're going to hear about erythropoiesis stimulating proteins. Oh no, we're probably going to do a motion, right? Thank you.
Oh, Lita, sorry. So it looks like actually for the last class, the oncology agents, we do have one of the products, it's in a class by itself. It's in the immune modulators, so we will actually need a motion for that product. Let me just double check. So I believe it's for the Revlimid, so we just need a motion for that one. I apologize.
This is Virginia. That's okay. I think that was that Marissa.
I'm sorry. Yeah, this is Marissa.
Okay, sorry. I guess we'll just wait then for Lita to give us some words to look at that.
Hang on, I'm sharing my screen. Let me check.
I can see your screen, Lita.
I can see it, Lita.
Yeah.
Okay.
Jenny, can you see my screen?
Yes, I can. Sorry.
Oh, okay.
So this is Susan Flattable. I just have a question. So Revlimid is in this class, but it's not, it has other indications though. I guess I'm confused as why it's in immune modulators, myelodysplastic syndromes, when it's also used for other disease states.
So that's just how we have it classified on the AHPDL. So of course, if there's other indications, then of course it can be used, because right now it does have PA for medical necessity. So if we do get any requests for them, if there is any FDA indications, it can still be used for the other ones. That's just how we have it classified on the PDL.
Okay, because it's also, I think it's used far more often in multiple myeloma and even for mantle cell lymphoma than for myelodysplastic syndrome. So anyway, I just found that interesting.
And Susan, this is Donna. I'm trying to figure out, trying to log in. It's possible that this is how Medispan classifies it. So I'm not 100% sure, or it is how Magellan classifies it. But I do think this is our drug class name. So we'll double check that.
Okay, thanks.
again, it's just one of those challenges when you have multiple indications that span across different disease states.
Well, I just wanted to make sure that, you know, it's still being approved for other reasons besides myelodysplastic syndromes.
Yeah, it would be approved for anything that it's indicated for.
So Donna, this is Connie. So do we need to get verification that this is the correct class before we go up through the motion? Or if we go through this motion, the default is that it'll already be approved for the classes it's indicated for? I mean, is there something that we need to finalize?
Which class it in, I don't think matters as much. It's in a class all by itself. So there's no preferred, non-preferred, you know, try and fail within this class if it's the only class, the drug in it. But we do cover everything for their FDA approved indications. And so it will, you know, regardless of what the drug class name says, if it has an indication that would put it into a different drug class, it would still be reviewed for that and approved when appropriate.
Okay. And so this motion currently is just specifically for the myelodysplastic syndromes. That is what we're reviewing. Is that correct?
Yeah, I'm not 100% sure. I think that it will be Revlimid in general. I did, going back to Umang's presentation, what was included in his presentation for Revlimid, do we recall?
Hey, Donna, it's Umang. Let me pull that up. It is Lita, slide 18. There we go.
Thank you.
This is Susan again. So pomalidomide or Pomalyst is essentially the same drug class as Revlimid, yet it's in, what was Pomalyst? What drug class is that in? I'm just surprised they're not in the same drug class.
Hey Donna, this is Amy. I just wanted to confirm the drug class is based on Medispan's classification, and they do not have the other product that was just mentioned classified in the same drug class.
So Susan, can you spell Pomalyst for me?
Yeah, it's Pomalyst. That's the brand name. The generic name is pomalidomide.
Okay, I'm looking at it right now, so hang on and I'll let you know.
I see it, Donna, in the oncology agents, immunomodulators, oral class on the PDL. And that's it's in a class by itself.
Yeah, I confirm that. It's just a matter of how Medispan is classifying the drugs. If you think that it's better that they be in the same drug class, we could move Revlimid into that anti-neoplastic immunomodulators class, but then it, you know, begs the myelodysplastic issue.
This is Susan again. I suppose it doesn't matter, just so long as that so they both pomalidomide and lenalidomide can be prescribed for multiple myeloma, and I don't want to feel like pomalidomide is necessarily preferred because they're in that immunomodulator class and lenalidomide is not. So does that make a difference if the doctor writes for lenalidomide? It's not going to matter, right, as far as preferred status over pomalidomide? Because they're essentially used in some of the same indications. Lenalidomide more first line, pomalidomide more second line. Yet the pomalidomide is listed in that immunomodulating class and lenalidomide is not. I don't know. I guess I'm fine with it as long as it's not raising questions about what they're being prescribed for.
I mean, they're both on prior authorization, so we would require the diagnosis. We're
Okay.
not preferring one over the other at this point. If they were in the same drug class, you know, they could potentially both be equally preferred, or we could prefer one to be used. You know, if you think that one should be used prior to another, that is, you know, we can do that through a prior authorization policy. But we can go back and just revisit, you know, what we call these classes as well. We have been trying to follow the Medispan because it's based on the what's called a generic product identification number, which identifies the drug, and Medispan has put them in two completely different therapeutic categories. So, but we can go back and look at, you know, the most appropriate place to put it. This isn't the first time this is, you know, the first drug class this has come up with.
Yeah, this is Susan. I suppose I'm fine with following however Medispan has them classified. Since they both require prior authorization, the oncologist would have all that information, you know, able to submit to get it approved. So I guess I'm fine with it. It just was kind of interesting to me that they classified them separately. But anyway, sorry. And then this is Susan again. One other thing about this slide, required trial of 2 preferred products. There may not be 2 preferred products, but I guess it says right in there unless or only 1 product is preferred. Never mind.
This is Virginia. Thanks, Susan. I just want to be sure, are we with the HCA, Donna, Lita, are we okay then to go ahead and consider this motion as it is?
Yes, that would be fine.
Okay.
Okay, so this is Connie Nguyen. I move that all products in the immune modulators myelodysplastic syndromes drug class are considered safe and efficacious for the medically accepted indications and are eligible for preferred status and grandfathering at the discretion of the HCA. Products in this class may require prior authorization to determine medical necessity. All non-preferred products require a trial of 2 preferred products with the same indication before a non-preferred drug will be authorized unless contraindicated, not clinically appropriate, or only 1 product is preferred.
Catherine Brown, a second.
This is Virginia Bucola. All those in favor?
Aye.
Aye.
The motion carries. And now we will proceed back to Umang for erythropoiesis stimulating proteins.
I think this is Marissa. I think we actually need to do the cytokine and CAM motion.
Thank you, Marissa.
No problem.
This is Virginia. I'm sorry. I heard you say that the first time and I got caught up in our discussion.
No, it's fine.
This is Susan Vladovo again. I just wondered if Umang could go back to the slide with the periodic fever syndrome. I don't recall what slide number it was. I just wanted to...
Let me find that for you. It is, Lita, it is slide 29, please.
So this is Susan again. My question is, so the Ilaris or the canakinumab, I just wanted to make sure that that was on there, and now I see it. Never mind. Okay, yeah, I guess I didn't notice that before, so that answers my question. And then I also have a, so as far as the biosimilars are concerned, where there's multiple biosimilars for some of these agents, does the HCA have a preferred biosimilar? Or is that even something that, you know, we need to discuss today?
Hi, Susan. So this is Donna. The biosimilars provide a profound challenge for Medicaid, as they are approved as a brand name drug. And oftentimes they are more expensive for Medicaid than their the original biological product that they are similar to, because of federal rebates and CPI penalties. For Uniform Medical Plan, the biosimilars are cheaper, and I think that cost is considered when Ryan is doing his analysis. So they might have, they might call out one biosimilar preferred over all other biologics that are of that same drug. So one etanercept biosimilar might be preferred, and then all other etanercept products be non-preferred. But that's done on a case-by-case basis.
Okay, thank you.
Yeah, they're not treated like generics, where, you know, like if it's a generic, they're all preferred. They are approved as their own brands, and they do have you know, significant cost differences.
This is Susan Vladovo. I move that all products in the cytokine and CAM antagonist drug class are considered safe and efficacious for their medically accepted indications and are eligible for preferred status and grandfathering at the discretion of HCA. Products in this class may require prior authorization for medical necessity. All non-preferred products require a trial of 2 preferred products with the same indication before a non-preferred drug will be authorized unless contraindicated, not clinically appropriate, or only one product is preferred.
This is Diane Schwilke. I second.
This is Virginia Bucola. All those in favor?
Aye.
Aye.
And the motion carries. I want to pause for a minute. This is Virginia. I'm assuming that the motion is carrying when I hear everyone say aye. So I just wanted to pause and acknowledge that if I need to formally ask if there are any in disagreement, I can change how I'm doing that.
This is Lita. Afterwards, you can say all of those opposed.
Okay, thank you. Okay, if there's anybody that needs to go back in time to this morning, we can send a message offline and we can do that. Thank you. All right, I think we are caught up then on motions. I keep trying to move us ahead too early, but I think we're ready to go back to Umang.
Okay. Perfect. Okay, so the next class that we will be reviewing is erythropoiesis stimulating proteins. And in this class, I will be doing much better about summarizing kind of the information, since I started out with the oncology and it was tough to kind of summarize cytokine and CAM. So with erythropoiesis stimulating proteins, to give a little background, anemia is a frequent complication affecting over 3 million people, Americans. It's associated with a number of serious diseases such as CKD, diabetes, heart disease, cancer, These conditions can cause anemia by interfering with the production of oxygen-carrying red blood cells. And sometimes, as in the case of cancer chemotherapy, anemia is caused by the treatment itself. So erythropoietin is a glycoprotein produced in the kidneys that stimulates the red blood cell production from bone marrow. Endogenous production of erythropoietin by the kidney is normally regulated by the level of tissue oxygenation, and hypoxia and anemia generally increase the production of erythropoietin, which in turn stimulates erythropoiesis. In normal subjects, the erythropoietin plasma levels range between 0.01 to 0.03 units per milliliter and may increase 100 to 1,000-fold during hypoxia or anemia. And in contrast, patients with CKD have impaired production of erythropoietin, which is a primary cause of the anemia. On the next slide here, just to give a little bit of background, there is a new sub-disease class that falls into this now, and is beta thalassemia. This is the first time since the last time we reviewed this class, so I will be a little bit more in depth with beta thalassemia. It's a rare inherited blood disorder marked by the reduction of functional hemoglobin levels, has an incidence of approximately 1 in 100,000 individuals in the general population. There are 3 subtypes of beta thalassemia, which are characterized by the severity of symptoms: minor, intermediate, and major. Individuals with beta thalassemia major require regular blood transfusions, as often as once every 2 to 4 weeks, and are dependent on medical care for survival. Treatment of beta thalassemia is highly dependent on the type of thalassemia, progression, severity, and the presence or absence of certain symptoms. Treatment options may include regular blood transfusion, chelation therapy, folic acid treatment, removal of the spleen and/or gallbladder, bone marrow transplantation. Reblozyl, which we will go over in a second, is the first FDA-approved erythroid maturation agent, which reduces patients' transfusion burden by regulating late-stage red blood cell maturation. It is approved for the treatment of anemia in patients with beta thalassemia who require regular red blood cell transfusions. On the next slide here, just to give you updates on guidelines in the last year, we have the National Comprehensive Cancer Network in 2020. Basically, they reinforced that physicians are advised to use the lowest ESA dose possible to maintain hemoglobin levels sufficient to avoid blood transfusion. They should be discontinued once the course of chemotherapy has been completed and anemia resolved in patients with cancer. And there's not enough evidence to support the use of ESAs for the treatment of anemia related to myelosuppressive chemotherapy with curative intent, patients receiving non-myelosuppressive therapy, or patients with cancer not receiving therapy. The ASCO, the American Society of Clinical Oncology, and the American Society of Hematology updated their 2010 recommendations for use of ESAs in patients with cancer. They emphasize the intent of treatment be considered when weighing the benefits and risk of these agents, including thromboembolism. And ESAs may be offered to patients with chemotherapy-associated anemia whose cancer treatment is not curative in intent and whose hemoglobin level is less than 10 grams per deciliter. They can also be used for low-risk myelodysplastic syndrome as well. And essentially, these guidelines reiterate that the goal hemoglobin should be the lowest value that prevents the patient's need for transfusion. And once that goal is achieved, ESA should be discontinued if there's a lack of hemoglobin increase by 1 to 2 grams per deciliter every 6 to 8 weeks. Going on to the next slide here, the new medication or updates in medication in the last year we have is Reblozyl. Its indication is it's an erythroid maturation agent indicated for the treatment of anemia in adult patients with beta thalassemia who require regular red blood cell transfusions, an anemia failing an erythropoiesis stimulating agent and requiring 2 or more red blood cell units over 8 weeks in adult patients with very low to intermediate risk myelodysplastic syndromes with ring sideroblasts, or with myelodysplastic myeloproliferative neoplasm with ring sideroblasts and thrombocytosis. The limitation for this is it is not indicated for use as a substitute for red blood cell transfusions in patients who require immediate correction of anemia. In terms of contraindications and warnings, it's similar to many other ESAs. Thrombosis or thromboembolism has been found. There's an increased risk in patients with beta thalassemia, and it is important for healthcare providers to monitor patients for signs and symptoms of thromboembolic events and institute treatment promptly. Hypertension, so monitor blood pressure during treatment and initiate antihypertensive treatment if needed. And lastly, embryo-fetal toxicity. It may cause fetal harm, and therefore healthcare practitioners are recommended to advise females of reproductive potential of the potential risk to fetus and use effective contraception. The dose is weight-based as well. And to give a little bit of background here, the mechanism of action is it essentially induces what I mentioned earlier, the erythroid maturation at the binding site, which increases the production of red blood cells here. In terms of pediatric patients, safety and efficacy has not been established. And for hepatic and renal impairment, there is no significant difference in mild or moderate hepatic or renal impairment, and there has been no evaluation for severe. That is the end of this therapeutic class. Any questions from the committee?
This is Virginia. I don't hear any questions. Is everybody ready to move on? Okay. It doesn't look like we have any stakeholders unless anybody's listening and has not put their hand up. Just want to give a moment for anyone to do that. Okay. And I'll presume that we can move to the motion.
This is Alex Park here. Just to clarify, when we talk about all the drugs in the class, we're not referring to the drugs that were in the slides. Is that right? We're talking about the list that was in the Magellan document?
Hey, Dr. Park, it's Umang. So when I do my slides, I've been informed when we revisit a class, I'm just reviewing the changes in the last year. So my slides would not be reflective of the entire class that the motion is discussing.
This is Alex Park. Thanks, Umang. That's helpful. So for the committee members to know what comprises the class, am I correct in assuming we are to refer to the other PDF? That's the Magellan document that has the whole list.
It would be referring to, this was Donna, referring to the Apple Health PDL. So let me look to see what is in this class.
Hey Donna, this is Marissa. So in this class, we have the Aranesp, Epogen, Procrit, Redicrit, and Mircera in this particular class.
This is Alex Park. Great, thanks. And that's looks like that's what's on page 2 of the other PDF that came with the documents for the meeting. Okay, I know where I'm looking for my information now. Thank you. This is Alex Park. I move that all products in the hematopoietic, I move that all products in the hematopoietic agents, erythropoiesis stimulating agents drug class are considered safe and efficacious for the medically accepted indications and are eligible for preferred status and grandfathering at the discretion of HCA. Products in this class may require prior authorization for medical necessity. All non-preferred products require a trial of 2 preferred products with the same indication before a non-preferred drug will be authorized unless contraindicated, not clinically appropriate, or only one product is preferred.
This is Connie Nguyen. I second.
This is Virginia. All those in favor?
Aye.
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 Any opposed? And the motion carries. And we will move back to Umang for colony stimulating factors.
Okay, thank you. So the next class that we have here is colony stimulating factors. Okay, to go back to a background, we have, so myelosuppressive chemotherapy can induce neutropenia. The definition is here, which, and febrile neutropenia, which is a dose-limiting toxicity of chemotherapy. The risk of febrile neutropenia is dependent on treatment and dose intensity, which is often underreported. Colony stimulating factors, or CSF, are hematopoietic growth factors that have been shown to decrease the likelihood of neutropenic complications resulting from chemotherapy and to improve relative chemotherapy dose intensity. Prophylactic CSF use can reduce the severity, risk, and duration of febrile neutropenia and decrease rates of infection and hospitalization. Medications such as Neupogen, Nivestim, Zarsio, Neulasta, Udenica, Fulphila, ZX-Tenzo, Granex are granulocyte colony stimulating factors, or GCSFs, and leukin is a granulocyte macrophage colony stimulating factor, or GM-CSF. On the next slide here, we have the NCCN 2020 practice guidelines for myeloid growth factors. In terms of these guidelines, safety data appears similar between Neupogen, Neulasta, and their biosimilars, and subcutaneous route is preferred for all agents. Subcutaneous filgrastim and its biosimilar Granex and pelfilgrastim have a Category 1 recommendation stating there is a high-level evidence from randomized control clinical trials, and there is uniform NCCN consensus that they prophylactically reduce the risk of febrile neutropenia. I apologize. I thought there was a question, but I think there may be an echo. Fulphila and Udenica have been designated a Category 2A. Due to recent approval, a recommendation of ZX-Tenzo is not currently provided by NCCN. To date, there are insufficient head-to-head comparative studies on the clinical benefits of GCSF and GM-CSF. Filgrastim, filgrastim biosimilars, and TBO filgrastim can be administered the day after chemotherapy up to 3 to 4 days after chemotherapy and through postnatal recovery. Based on data from clinical trials, pegfilgrastim and its biosimilars should be administered the day after chemotherapy. However, administration up to 3 to 4 days after chemotherapy is also reasonable according to the NCCN guidelines. For patients unable to return to the clinic the next day for medication administration, a delivery device such as the Neulasta Onpro is available that allows for the device to be applied to patients the same day as chemotherapy administration, but the device does not release the medication until approximately 27 hours after the application. There is evidence to support the use of chemotherapy regimens every 3 weeks with pegfilgrastim and its biosimilars. Efficacy data exists for pegfilgrastim products and chemotherapy regimens given every 2 weeks. There is insufficient data to support dose and schedule of weekly chemotherapy regimens, therefore pegfilgrastim products should not be used. And leukin is no longer recommended for prophylactic use in patients with solid tumors receiving myelosuppressive chemotherapy. On the next slide here, continuing with the updated NCCN guidelines, the guidelines note that a biosimilar is a biological product that is highly similar to the FDA-approved originator product with very small clinically inactive differences, but no difference in efficacy, safety, or purity. The first biosimilar, Zarsio, was approved in March 2015 with a second filgrastim biosimilar, Nivestim, being approved in 2018. However, Granex was approved in 2012 as a biologic rather than a biosimilar in the United States. In Europe, Granex is available as a biosimilar to filgrastim. The first pegfilgrastim biosimilar, Fulphila and Udenica, were approved in 2018. Studies have shown these products have similar safety and efficacy profiles as the originator product. The guidelines state if overall safety and efficacy are equivalent, biosimilars may be substituted for the originator product. However, the guidelines note that current biosimilars are not interchangeable. Therefore, alternating between a biosimilar and its originator is not recommended. The guidelines also note that the use of biosimilars may provide opportunities for cost containment. The guidelines endorse the use of Zarsio, Nivestim, Granex, Fulphila, and Udenica for myelosuppressive doses of radiation. For mobilization of hematopoietic progenitor cells in the autologous setting, the use of concurrent filgrastim or one of its biosimilars with sargramostim or single-agent filgrastim, Zarsio, Nivestim, or Granex is recommended. Filgrastim, Granex, or filgrastim biosimilars are all NCCN 1 Category 1 GCCF options for prophylaxis of febrile neutropenia and maintenance of scheduled dose delivery. According to the guidelines, the World Marrow Donor Association recommends pegfilgrastim or filgrastim biosimilars for the mobilization of peripheral blood progenitor cells in healthy donors in allogenic settings. The guideline does not recommend Neulasta or its biosimilars for mobilization at this time. Here on the next slide, you'll see a new medication, ZX-Tenzo. In November 2019, FDA approved this medication. The indication that it is approved for is it is a leukocyte growth factor indicated to decrease the incidence of infection as manifested by febrile neutropenia in patients with non-myeloid malignancies receiving myelosuppressive anti-cancer drugs associated with a clinically significant incidence of febrile neutropenia. So there is a limitation. It is not indicated for the mobilization of peripheral blood progenitor cells for hematopoietic stem cell transplantation. In terms of contraindications and warnings, patients with a history of serious allergic reaction to human granulocyte colony stimulating factors, such as pegfilgrastim products or filgrastim products, fatal splenic rupture have occurred. Evaluate patients who report left upper abdominal or shoulder pain for an enlarged spleen or splenic rupture. Acute respiratory distress syndrome has occurred, so evaluate patients who develop fever, lung infiltrates, or respiratory distress, and discontinue in patients with ARDS. The dosage is here. Again, it is also semi-weight based. And in terms of mechanism of action, this medication is a colony stimulating factor that binds to specific cell surface receptors and stimulates proliferation, differentiation, commitment, and end cell functional activation. In terms of patients who are pregnant, there's limited data in pregnant women, and there is no identified drug-associated risk for miscarriage or major birth defects. And in terms of pediatrics, it is approved for pediatric patients and has specific dosing instructions for patients less than 45 kilograms. That is all I have for the colony stimulating factors section. Any questions from the committee?
This is Virginia. It doesn't sound like there's any questions. Thanks, Umang. We have one stakeholder, Jennifer Shear from Teva Pharmaceuticals. If there are any others, Jennifer, you'll have 3 minutes to share your comments.
Thank you. Can everyone hear me?
Yes.
Great. Good afternoon. My name is Jennifer Shear. I'm a medical outcomes liaison with Teva Pharmaceuticals. Today, I'd like to provide you with a short update on Granix and its pediatric data. First of all, Granix TBL filgrastim is indicated to reduce the duration of severe neutropenia in adults and pediatric patients 1 month and older with non-myeloid malignancies receiving myelosuppressive anti-cancer drugs associated with a clinically significant incidence of febrile neutropenia. In terms of pediatric use, the safety and effectiveness of Granix has been established for pediatric patients 1 month to under 17 years old. The use of Granix in these age groups is supported by evidence from adequate and well-controlled studies of Granix in adults. Additional safety and pharmacokinetic data was obtained from a phase 2 multi-center open-label trial that investigated the safety and tolerability of Granix in pediatric patients receiving myelosuppressive chemotherapy. In total, 50 patients aged 1 month to below 16 years of age with solid tumors without bone marrow involvement were prophylactically administered TBL filgrastim 5 micrograms per kilogram body weight, once daily subcutaneously. Granix administered to pediatric patients demonstrated a safety profile consistent with the safety profile in that of adult patients. The incidence of febrile neutropenia was on the lower end of the range reported in literature, and the incidence and duration of severe neutropenia provided supportive data on the efficacy of Granix in pediatric patients that are in line with previous research. And this concludes my comment today, and I'm happy to answer any questions.
Thank you, Jennifer. Any questions from the committee for Jennifer? Okay. Lita, I see that it looks like there's another stakeholder.
Good afternoon. My name is Govind Shah. I'm a medical science liaison. On behalf of Coherus, I thank you for the opportunity for providing this comment pertaining to Udenyca, pegfilgrastim CBQV, a biosimilar to Neulasta pegfilgrastim. Udenyca is a leukocyte growth factor indicated to decrease the incidence of infection as manifested by febrile neutropenia in patients with non-myeloid malignancies receiving myelosuppressive anti-cancer drugs associated with a clinically significant incidence of febrile neutropenia. UDENYCA was approved by the FDA as a biosimilar to Neulasta on November 2nd, 2018. The clinical development program for UDENYCA included the following: 1, extensive analytical data; 2, preclinical data; 3, key confirmatory UDENYCA clinical studies. The first of that clinical studies was CHS 1701-05, a confirmatory PK/PD bioequivalence study with a 3-sequence, 3-period crossover study design in 122 healthy subjects that demonstrated PK and PD bioequivalence between UDENYCA and Neulasta. Healthy subjects are the FDA preferred population due to the lack of confounding variables for PK and PD evaluation. The second study was CHS 1701-04, which is a 2-period parallel arm immunogenicity study in 303 healthy subjects that demonstrated similar immunogenicity between UDENYCA and Neulasta. There was also a pooled safety analysis in which no new important safety signals were observed in the clinical studies conducted for the UDENYCA program. The most frequently reported treatment emergent adverse events in both treatment groups included bone pain, back pain, headache, pain in the extremity, and arthralgia. In conclusion, the FDA did not require Coherus to perform clinical trials in cancer patients due to the strength of the bioanalytical data and the results of the 2 clinical trials in healthy subjects. UDENYCA was FDA approved. Of note, we have delivered over 150,000 doses and over 55,000 patients have received UDENYCA, and the safety profile is consistent with pegfilgrastim products. In addition, with the healthcare market entry of biosimilar pegfilgrastim as a clinical option at a lower price, as well as a deterrent to originator price increases, UDENYCA has contributed to nearly $1 billion in cost savings. In conclusion, I would like to thank the committee for the opportunity to present this testimonial, and I would be happy to take any questions.
This is Virginia. Thank you, Mr. Shaw. Are there any questions from the committee? Okay. We are scheduled to move to a break, but I think because of some timing issues, we need to jump ahead right now and go to Apple Health Policy, androgenic agents, testosterone, I believe. Oh, I keep forgetting to do the motion. My apologies. Let's back up and do that. Thank you.
This is Nancy. I move that all products in the hematopoietic agents, granulocyte colony stimulating factors drug class are considered safe and efficacious for their medically accepted indications and are eligible for preferred status and grandfathering at the discretion of HCA. Products in this class may require prior authorization to determine medical necessity. All non-preferred products require a trial of one preferred product with the same indication before non-preferred drug will be authorized unless contraindicated or not clinically appropriate.
This is Alex Park, I second.
This is Virginia Bucola, committee chair. All in favor?
Aye.
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 Aye. Any opposed? That the motion carries. And
Ginny, this is Lita.
Hi, Lita.
I just wanted to say we can take a 10-minute break. Sickle cell anemia is canceled. And then when we come back from break, we're going to move androgenic agents, testosterone, above the oncology radiopharmaceuticals. Just change the order of those two. So we're doing okay on time.
Okay, good. So let's go ahead then and take a 10-minute break. It's 2:15, so I'll see you all back at 2:25.
Great, thank you.
Hi, this is Virginia, committee chair. Welcome back from break. And we're going to go to Ryan Pistresi with HCA and talk about androgenic agents.
Great, well, thank you. So this is Ryan Pistresi from Healthcare Authority, and we will start with the first of the clinical policies that we'll be presenting today. So we're at the home stretch for this meeting. So let's see. So this is a policy that has been previously reviewed by the DUR board, and this is up for re-review today. I wanted to just walk through some of the changes with this policy to kind of help guide your eyes as you look through it. It's about several pages long, but I will point out some of the key areas where it's changed. If we are able to scroll down to that medical necessity section, you will see that we have updated the list of drugs to the left to reflect what's on the market. And we've also better reformatted the medical necessity table to the right. So you'll see that it's better listed for kind of what section that you would find that criteria in. So if you're looking for how testosterone would be approved for use in HIV-associated weight loss, you can see there it would be in the testosterone replacement therapy section. I do want to draw your attention to the criteria for gender dysphoria down at the bottom of that table. As you see, there is a note there that says that the use for testosterone is actually moving to a transgender health policy that will be scheduled for an upcoming DUR board meeting. So we do not have new criteria to share with you today, but we will be bringing that to you at a future meeting. This move allows us better flexibility in updating the medications for gender dysphoria together and to align with some of the other programs at HCA. So rather than updating, several clinical policies at once, we'll have that all together in one policy, and so we'll be bringing that for a future meeting. If we continue to scroll down, now we'll actually be getting into the clinical criteria that we'll use to review a medical determination. So the criteria for each of these policies is mostly the same as from the previous policy that you reviewed and approved, but we are making clarification about what documentation we will be required for us to do a prior authorization review. So in this first section, you can see we have the testosterone replacement therapy for adult males. And this was formatted to better describe what criteria would be used for the different indications. And that can be found in criteria number 4. If we can scroll down, you'll see that each of the diagnoses here have the different criteria in them. So if you're looking for the criteria that we would use for primary hypogonadism, you'd be looking at 4A, and you would not need to look at 4B or C or any of the other sub-bullets to criteria 4. So the criteria is largely the same, just updating the language to make it clear what documentation would be required by Apple Health to make a medical determination for appropriate use. Okay, we can scroll down to the reauthorization criteria section. So yeah, I guess it's kind of split between these two pages. So let's kind of scroll down to the next one actually, because that's where most of the, great, thanks. So for this reauthorization section, it's to verify, response to treatment and including specific criteria now for chronic high-dose glucocorticoid therapy and on the HIV-associated weight loss. So better describing what we're looking for here to ensure that these patients are responding to therapy and that reauthorization is necessary for the current treatment of their condition. So like for the chronic high-dose glucocorticoid therapy, just letting us know that they are still continuing the therapy and that the testosterone is necessary for that patient. If we scroll down, so the next section will be on delayed puberty. Thank you, Lita. So this was updated to clarify when authorization would be approved, because previously it would only look at the initial authorization criteria. So most of the updates for delayed puberty were in the reauthorization section. But the formatting had been updated to make it clear exactly what labs and what criteria would be necessary for that initial determination. And so there, and that reauthorization section at the bottom had been updated. Okay, below this is the metastatic breast cancer section. Okay, thank you. And so this one, we removed the recommended therapy section and instead just focused it on the clinical criteria like we have for our other clinical policies that we've presented. Otherwise, there wasn't really any changes besides general formatting. Okay. And if we continue to scroll down, you'll notice that we don't have the transgender health criteria anymore, as that is being moved. So now it's going to be reviewed at a upcoming DUR board meeting. So this ends the clinical criteria for the proposed testosterone policy, and everything that you see from here on out is more general information about the drugs that are covered by this policy. So here is the updated list of drugs and the quantity level limits, which you can find in the right column. And then below that is our relevant HCPCS codes, the references that we use to develop this policy, and then at the very end is the history of this policy, which we are using to document what changes have been made between the last time this was reviewed in 2018 to what we have here today. So bringing this back to the board, do you have any questions, comments, thoughts on the, oh yes, thank you. So this is the pen form that we are proposing for the testosterone PA. So this is the pen form that would be used, that we would send out to gather the information for the prior authorization determination. Sorry, I forgot we had that at the end. And so this just helps organize the data from the clinical policy that we had just presented and shows you kind of how we would be asking for it from a provider's office. So now back to the board. So any questions, comments, any feedback for HCA on this testosterone policy?
This is Virginia. Thanks, Ryan. Anybody have any questions that they needed to ask?
Okay. Yeah, I just have a quick question. So when you say that the February 2020 update for transgender health is included, it means that it's going to be included in a separate policy. Is that correct? Because I thought that's
yes. Yes, we have, sorry, so this is Ryan. Yes, we do have staff that are currently working on the transgender health clinical policy, and we are planning on bringing that to an upcoming DUR board meeting, but it is not ready for today.
Excellent, thank you.
Thank you.
This is Nancy. All I just wanted to say was thank you. It looks good.
Great, thank you.
Okay, this is Virginia. It doesn't look like as if we have any stakeholders, and so we'll move right to the motion.
This is Nancy. I move that Apple Health Medicaid program implement the clinical criteria listed on policy 23.10.00 as recommended.
This is Susan Guadabo. I second.
This is Virginia Bucola. All those in favor, please say aye.
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 Any opposed? And the motion carries.
Great, thank you.
Thank you. So we're going to move next to Luke, HCA, to discuss oncology radiopharmaceuticals, specifically Lutathera, I believe.
Yes, thank you. Can you hear me?
Yes, I can.
Great. So my name is Luke Dearden from HCA, and the purpose of this is to discuss Apple Health policy regarding luteum dotate, also known as Lutathera. And unlike the testosterone policy, this is a new policy to Apple Health. So a little bit of background: Lutathera is a radio-labeled somatostatin analog and was approved by the FDA in 2018 for the treatment of somatostatin receptor positive gastroenteropancreatic neuroendocrine tumors. In the NETTER-1 trial, which is summarized at the bottom of this document, Lutathera in combination with standard of care was compared to standard of care alone, which happened to be long-acting octreotide. Lutathera contributed to a progression-free survival at 20 months of 65.2% compared to 10.8% in the standard of care group, which was statistically significant, and that was the primary outcome. There was an interim analysis of overall survival that revealed 14 deaths in the Lutathera group versus 26 deaths in the standard of care group. However, final analysis of that is ongoing and will be concluded after either 5 years or 158 total deaths. The overall population of that trial was 229 patients. Lutathera is administered intravenously once every 8 weeks for a total of 4 doses. And the clinical criteria, if we scroll down here, they're largely based on, they're largely based on the phase 3 trial that I just described, the product labeling, and also the National Comprehensive Cancer Network, or NCCN. So I'll provide a very high-level overview of the criteria. They include appropriate diagnosis. The tumor must be well differentiated, must be inoperable or metastatic. There must be documentation of disease progression on standard of care, like I said before, which would be a long-acting somatostatin analog. The client must have lab work that demonstrates adequate renal and bone marrow function. It must be over 18 years or older, cannot be pregnant or breastfeeding. Somatostatin analogs must be discontinued prior to Lutathera administration. And this policy has a maximum lifetime quantity of 4 doses. So at this point, I guess if you scroll down a little bit more, just kind of goes over what I summarized. And then the dose and quantity limits down below, which summarizes the 4 maximum doses and also gives a summary of how it is supposed to be appropriately administered. And below that is just a summary of the background and clinical trials, and also a brief summary of the safety data with this medication. So I guess I would direct it back to the committee now if there are any questions.
This is Virginia. Thank you, Luke. Want to make sure I'm not talking over anyone who may have had questions. Okay. I don't see that there are any stakeholders, so we will go ahead and move to the motion.
So just one other thing, this is the pen form as Ryan was previously talking about. This is the information we collect from the clinic to make sure this policy or the medication prescribing is in compliance with the policy.
This is Susan Flattabo. I move that the Apple Health Medicaid program implement the clinical criteria listed on policy 21.60.00.45.20 as recommended.
This is Jordan Starhaug. I second.
This is Virginia Piccola. All those in favor?
Aye.
Aye.
Any opposed? And the motion carries. All
Great,
right.
thank you.
Thanks a lot, Luke. I believe next, I'm following things along correctly, we should be with Marissa to discuss endocrine and metabolic agents. And I'm just going to use the brand name with Recovi.
Yep, thank you, Jenny. Can you guys hear me okay? Let me actually turn on my webcam.
I can hear you. Thanks.
Okay, great. I was having internet issues earlier, so hopefully everything goes smooth. So today I'll be presenting our clinical policy, our proposed clinical policy for Revcovi. So just to give you a bit of background on Revcovi and the disease state that it treats. It's for adenosine deaminase deficiency, specifically severe combined immune deficiency. So just for simplicity's sake, I'll call it ADA-SCID. The drug Revcovi is a recombinant adenosine deaminase, and currently, or before, there was a product that was made before Revcovi, which is called Atigen, and that product is called pegadeaminase. But because Atigen is actually discontinued due to the permanent shortage of I'll just call it Revcovi. Now Revcovi is replacing Atigen. So we'll go ahead and scroll down, Nita. Oh, that's perfect. So Revcovi, the clinical criteria here that we've created are based off of the phase 3 open label one-way crossover study. So the study that was conducted, there was a crossover where patients were first started on Atigen,
and then they were given Revcov and then continued on Revcov. So the criteria was based off of the methods for that particular study. And the primary endpoint for that particular study was measuring trough DAXP levels, and that's having a trough DAXP less than 0.02 millimoles per liter. And it was found that about 5 out of the 6 participants in the study were able to reach that particular endpoint. And then also having another endpoint was plasma ADA activity greater than 15 millimoles per hour per liter. But it was found that when participants were given Revcov, their plasma ADA levels were actually, they maintained an average of greater than 30. So that's actually included in the criteria here. So just to get into some of the criteria that we have created, they have to have a diagnosis of ADA-SCID, which is confirmed by genetic testing, or if they have very low ADA catalytic activity, which is less than 1% of normal than their baseline. Per the package insert of Revcov, it's not recommended for patients to use this if they have severe thrombocytopenia. And for ADA-SCID, there is a consensus statement made by the Journal of Allergy and Clinical Immunology, and they do recommend that the treatment for this is specifically stem cell transplant. So ERT is really used for bridging, or if they're not candidates of HSCT at the time or have failed HSCT. So that is one of the criteria included here. Go ahead and scroll down, Lita, please. And then also if it's prescribed and administered by a specialist or physician in ADA-SCID, and then also monitoring, so monitoring the trough plasma ADA activity, the trough DAXP levels, lymphocyte counts, and neutralizing antibodies. And then the reauthorization criteria is listed below. And then if you want to scroll down some more, Lita. So, go ahead. Yep, here's the dosing. So this is the dosing for Revcov specifically. So like I said, Atigen was discontinued, but I did find that it was listed that Atigen had an obsolete date of June 20th of 2020. So there could still be, they did think that Atigen was going to run out in March of 2019, but I did see that there was an obsolete date of June 20th, 2020. So there could still be patients that were on Atigen. And on the package insert of Revcov, they do have dosing specifically for if patients were started on Atigen and transferred over to Revcov or transitioned over to Revcov. This is the dosing schedule that they would follow. But there's also dosing for if clients are not if they are Atigen naive. So that is listed here as well. And then go ahead and scroll down, Lita. And then scroll down. I think if you can go to the pen form. And then here is the authorization form where we collect the information from the providers if they're requesting Revcov. And it is in line with the criteria for the policy. So if the board has any questions, we can review it, and please let me know.
Thanks, Marissa. Any questions from the committee for Marissa? Okay, we have one stakeholder. I see Tom Arnhart representing Ultragenics. Tom, if you're ready to go, you'll have 3 minutes to share your comments.
This is Lita. I do not see Tom on the attendee list. So Tom, if you are on, please raise your hand. You might have come up under a different name. Okay, I see here under Megan Bell. And unmute yourself. There you go.
Hi, good afternoon. Hey, can you hear me okay?
Yes.
Yes.
And so I'm speaking on Chris Vita under the endocrine and metabolic agents. Is that the category we're speaking on now?
This is Virginia. No, that is our next category.
Right, so I'll be speaking on the next category then. Thank you very much.
Sure, we'll get back to you then. Thanks.
Thank you.
Okay, so let's go ahead then and move to Ryan Takatomo with HCA to talk about endocrine and metabolic agents, specifically Chris Vita.
Hey, Jenny, this is Marissa. I think we actually have the motion for the Revcovy.
Why can I not remember to Thank you so much.
No problem.
Do the motion. It's right there on the agenda. It's not like it's something different.
It's Alex Park here. Marissa, can I ask you a quick question?
Yes. Hi.
I definitely won't pretend to be an expert on this condition, and so I've got my little drug database situation opened up here, and I've been following along with you, and the criteria appears quite appropriate based on the reading that I've been doing as you've been presenting. The only question I have is the reauthorization. So number 4, it says that the prescriber verifies client is still not an eligible candidate. So what if they have failed definitive therapy with stem cell transplant? Would that be considered being not eligible?
For Revcovy or for stem cell?
For Revcovy.
Sorry, hold on. I'm trying to look at you said it was number 4 in the reauth?
So,
yeah.
Alex, this is Donna. Are you asking if they've had a stem cell transplant and then they relapse, are they then eligible for Revcovy?
Yes, I just want to make sure that patients can still have access to it if they've had definitive therapy with stem cell but failed. That's part of the original initial approval criteria, but I don't see it in the reauth criteria.
We can add that in.
Okay, thank you.
Yeah, I think that should be included in the reauthorization criteria.
Okay, great, thanks.
And do you want to tell me again, was that up? This is Vita. Can I copy and paste it in?
I think you could, let's see, take from number 3 under initial approval and make number 4 under reauthorization match that.
Yeah, I think, Alex, this is Donna again. I think that's what the intent of number 4 was, but to explicitly state it is much more clear.
Thanks, Donna.
I'm going to open up the Word.
Vita, I am documenting it on a Word document as well, so I have it.
Oh, okay. All right.
Yeah.
This is Alex Park, and then in terms of the motion, so is there a difference in the policy that's listed here versus the one that's in the policy that we just reviewed? What am I trying to say? There's a -1 on the policy that we're seeing here. So I guess that must be a more recent version that we're trying to approve.
This is Donna. That's just the version number. So it's this policy is the number, and then the -1 would be the version. It's version 1.
Okay.
Actually, Lita, can you put the -1 after the last 30? Is that better, Alex?
Thanks for placating my OCD.
No, it's fine. I think we should specify that it's version 1 as well. So that was very helpful.
Actually, on that topic, I think there was a -2 on the androgen policy that we might have to update, Lita. We could probably take care of that on the back end. Beautiful. Okay, with that, this is Alex Park. I move that the Apple Health Medicaid program implement the clinical criteria listed on policy 30.90.20.30-1 as recommended.
This is Diane Schwilke. I second.
And this is Virginia Bucola. All those in favor?
Aye.
Aye.
Any opposed? And the motion carries. Okay, moving on. Now we're ready to go to Ryan Takatomo on talking about Crysvita.
Good afternoon, everyone. Can you hear me?
Yes, I can hear you, Ryan.
Thank you. All right, so we'll be talking about the Crysvita policy. This was a policy previously approved, and so for this annual review, we will be discussing a few changes which have been made, which incorporate new evidence and feedback which we have received. So a brief overview of the background. Crysvita is used to treat X-linked hypophosphatemia, which is an inherited disorder which affects 1 in approximately 20,000 and is caused by a mutation in the PHEX gene. This mutation increases fibroblast growth factor 23, which reduces reabsorption of phosphate by the kidneys. Symptoms of hypophosphatemia include rickets, bone softening, muscle pain, bowing of legs, short stature, tooth abscesses, and hearing loss. Symptoms for this disease are also highly variable, as some patients show no signs or symptoms and others may experience complications. Typical treatment for X-linked hypophosphatemia has for a long time been phosphate supplements with calcitriol, and so with burosumab or Crysvita, burosumab works by binding to and inhibiting fibroblast growth factor 23, which restores renal absorption of phosphate. So now we can move on to the policy section, or the criteria section, where I will highlight some of the big changes. So with criteria 1, we expanded the age range to reflect new FDA labeling, which reduces the age limit from a minimum of 1 year to 6 months of age and older. With criteria 2 and 3, they are mostly the same. We added, from feedback we received, that we wanted to add some additional guidance around the laboratory values for criteria 2C. So if they don't meet 2A or 2B, they can use 2C. And pretty much these values are not to be definitive, but are to act as guidance in that patients with this disease, depending on what stage they're at, they may or may not have an abnormal laboratory value. So the values are really for guidance based on the feedback we have received. Criteria 3 and 4 are same as the previous version. With criteria 4 and 5, these were split out. They used to be combined into one line. However, we received feedback that it was a little bit complicated to read, and so those have been split out. And 6 and 7 are the same as the previous version. So we can move down to the reauthorization criteria, please. Okay, so with the reauthorization criteria, points 1, 2, and 5 are the same as the previous version. The only changes were 3 and 4, which is that the client is not taking an oral phosphate or vitamin D, as these are contraindicated. And so with that, we have the dosing and quantity limits, the HCPCS coding, some definitions, and then a review of the clinical trials. And then Lita, if you could go to the end. And here we have the pen form, which we use to facilitate the prior authorization process. So with that, please review the policy. If you have any questions or feedback, please feel free to ask those.
Thanks, Ryan. Any questions from the committee for Ryan? Okay, and again, this is Virginia. And now we do have the one stakeholder that we introduced with the last topic, Tom Arnhart from Ultragenics. Tom, you'll have 3 minutes to share your comments.
Tom, there you go.
Hi, good afternoon. I'm Tom Arnhart. I'm a pharmacist with Medical Affairs and Ultragenics, and really didn't have a public comment I want to make. I just wanted to point out a clarification with Crysvita or Brosmab for XLH in use in adult and pediatric populations. As you mentioned, you updated your policy for the recent changes from the FDA. So I wanted to clarify that in the policy on the dosage, the range down to 6 months of age, on your policy, it shows the dosage as 1 milligram per kilogram, rounded to the nearest 10 milligrams. That is the adult dose. With that change back in September of last year, there was a difference for the pediatric dosing. For patients who weighed less than 10 kilograms, the recommended starting dose was 1 milligram per kilogram, rounded to the nearest 1 milligram. And for patients who weighed 10 kilograms and greater, the recommended dose was 0.8 milligrams per kilogram, rounded to the nearest 10 milligrams. So I just wanted to clarify that. And also wanted to bring up one other thing to your attention, that Ultragenics recently announced that the FDA had accepted our supplemental biologic license application for Crysvita for an ultra-rare disease called tumor-induced osteomalacia. It was assigned a priority review with a target PDUFA date of tomorrow, June 18th. So I just wanted you to be aware of that potential new indication. So appreciate your time and happy to address any questions.
Thank you, Tom. Any questions from the committee? Okay, we'll move to the motion.
Vita, this is Marissa. Can you add a -2 after the 95? Thank you.
This is Alex Park. I move that the Apple Health Medicaid program implicate the clinical criteria listed on policy 30.90.95-2 as recommended.
Catherine Brown, I second.
This is Virginia Bucola. All those in favor?
Aye.
Aye. Any opposed? The motion carries. And we will move again back to Ryan to discuss agents for ALS, Radicava.
Okay, so for the next policy, we have edaravone or Radicava. A brief overview of this drug background. This medication is used to treat amyotrophic lateral sclerosis, also known as Lou Gehrig's disease. It's a progressive disease that causes degeneration of motor neurons. Symptoms of this disease initially present as slurred speech and dysphagia, but as the disease progresses, patients may lose their ability to control muscle movement and may eventually develop paralysis. So with this drug, Radicava's current mechanism of action is not known, but it is thought to be due to its antioxidant properties, which prevent oxidative stress to neurons. Okay, if we could please move down to the clinical criteria. And so the clinical criteria for this policy was based off of evidence and FDA labeling. I can provide a brief overview of the trials that kind of went into this criteria. So there are 3 trials. The first trial was an initial trial that had 206 subjects with ALS. And their primary outcome was looking at the change in ALS functional rating scale, the revised version. And at 24 weeks, they found that there was no significant difference between edaravone compared to placebo. However, they conducted a post hoc analysis which showed that a particular subgroup of subjects with definite or probable ALS and other criteria did seem to have some benefit. So they conducted this second trial. This second trial enrolled 137 patients with early stage ALS. And this criteria was more specific as it was a subgroup. It targeted patients who had a disease duration of 2 years or less. They were living independently. They had a score of 2 or more on all items of the ALS-FRS revised scale, and they also had a forced vital capacity greater than or equal to 80%. At the 24-week mark, they did find that there was a clinically significant difference with decreases of -5 and -7.5 for Radicava and placebo, respectively. And this difference was considered to be clinically significant with a slowing of approximately 33%, and that's relative. And so the third trial was sort of a long-term extension to kind of gain some additional data. There was no statistical analyses involved with this trial. And so what kind of happened was the participants from the second trial I discussed, they continued, and the members who were on placebo were given Radicava. At the end of the study, they found that both groups continued to have decreases in ALS-FRS revised scores, with the ultimate conclusion that long-term treatment for efficacy and safety remain a future issue. And those are the kind of summaries of the clinical trials that went into the development of this criteria. So with that, we then go into sort of the other general information with the dosing and quantity limits, the HCPCS coding references, and at the end, there should be the pen form with the criteria that helps facilitate the prior authorization process.
Ryan, this is Virginia. Are you ready for us to move to questions, or did you need more?
Oh, I was wondering if it was lagging because I wasn't seeing the document scroll, but sorry, I should have
No,
maybe specifically stated to scroll down. Is there a pen form for this one, Lita? And so with that, now we can open up for questions and feedback.
This is Virginia. Thanks for that, Ryan. Committee members, any questions? Okay. I see that we have one stakeholder listed, and I do see a hand raised. So the first stakeholder is Bill with Mitsubishi Tanabe Pharma America.
Yes, can you hear me? Okay.
Yes, Bill, you have 3 minutes.
Great, thank you. Ryan, thanks for the summary. My name is Bill Gittinger. I'm the Director of Government Accounts, which is Medicare and Medicaid for Mitsubishi Tanabe Pharmaceuticals. We have one comment on the policy as it's drafted here, and if it's possible to go back up to the criteria for reauthorization, please. There, thank you. We see this reauthorization, number one, frequently with payers. It's common. Just to level set here, though, these patients do not get better. They're ALS. The Radicava simply slows the loss of function, but ALS patients are, Radicava is the last product for them, more than likely. And using the ALS FRS scale to determine, that they should be on it is great. But these patients will all still slide as they go on, and it's requiring a score of 2 or better on each of 12 boxes on the scale. And if any, as it's written here, if any patient drops to a 1, even though they're out, walking to get the paper or walking to get the mail, if they drop to a 1, they're no longer eligible to receive Radicava. And what we recommend to payers are 2 recommendations or 2 options, is to, instead of having this language, use the language for medical necessity. And we recommend that the decision to continue or discontinue be between the doctor and the patient. Does the patient want to continue to fight the good fight, or do they not feel it's worth it anymore and they're going to accept their position? And then we have some payers that will say, Bill, we really like the idea of a score, but we don't want to penalize a patient for dropping on one area from a 2 to a 1. The recommendation is theirs to give the patient a cumulative score and then to set a drop percentage on that score. And what payers are doing is they're looking at it and saying, okay, as long as the patient doesn't drop more than 4 points, then they can stay on if they choose to. But to have them thrive or to do well on 11 and then have just one area knock them out, again, we think that maybe they go from a 25 to a 20, that Radicava is probably not working well for them. But if they go from a 25 to a 23, they're probably still doing well and can continue to do well on the product. So we would like to have you just consider modifying this language and making it between the physician or a patient, or do a cumulative. And I think that's my time. So I would be glad to answer any questions or provide follow-up material. So thank you.
Thank you, Bill. Are there any questions from the committee for Bill? Okay, we will go ahead and move to the motion.
So this is Connie, just a quick question. Are we missing any dashes since we had dashes before for the policy?
So if we were to add dash, it would be a -1 for this one.
Yeah, this is Marissa. We can add that to the policy as well. I don't believe it has a -1, but we'll add that.
It's Alex Park here. Can I ask you a question, Ryan?
Sure, go ahead.
So, I mean, you characterize quite thoughtfully, I think, the data for this seems to be best in early ALS, and I'm guessing that's why the policy says onset of ALS less than or equal to 2 years. But, I mean, this is a pretty devastating disease, and so what happens if a patient is at 4 years and they want to try this?
And so that's sort of the struggle where we had to balance access with evidence that is currently available, because as we talked about, or as I talked about the first trial, where the onset of the disease for that one was up to 3 years, they found no significant difference between Radicava and placebo.
And hi, Dr. Park. This is Donna. I'm gonna jump in here. So, you know, based on the FDA indication, it would be approved. You know, it would not be. It would be reviewed on a case-by-case basis for anything outside of the policy. And if it were approved, I think that's where relying on the scores for the reauthorization would be extremely important to make sure that the patient is actually improving on the medication rather than just staying the same or getting worse, continuing to get worse. So there is a way to get to the medication for patients that are have had ALS for more than 2 years.
This is Alex Park. Well, that's reassuring. I just want to make sure the policy jives with that, because when I read this, it says, you know, number 1 and number 2 and number 3 and et cetera, et cetera. And it kind of reads like you have to have that time limit on the 2 years or less. Was that not how you intended the policy to read?
I think that is intended. I think what I was talking about was more as an exception or a reconsideration of, you know, is there a reason why they really think the medication would work in this particular patient that was above and beyond the 2-year mark of ALS.
Okay. Thank you.
This is Leah. I had a quick clarification as well. So related to the stakeholder comment, are there policy exceptions that can be made for patients in the example that there is a single point score difference, but in other in some of the other dimensions, there's either improvement or evidence that there is effectiveness of the medication, or is that the way the policy worded going to restrict people potentially in those situations?
I think we could take into consideration the point from the stakeholder regarding not falling below 2 in all 12 areas of the testing. And, you know, if you want to make some recommendations on what, you know, is there a number that you feel comfortable putting in there, or just, you know, maybe it's, you know, they don't fall below 2 on, you know, more than 2 of the 12 or 3 of the 12 areas.
I mean, I would probably want some specialist input for people who are prescribing this to answer that question. But I think that would be helpful just to make sure that the policy isn't so restrictive that we're reducing access to people who may benefit.
So we can take that back and you know, get some further review from providers that treat people with ALS and see if we can come up with a recommendation for the reauthorization criteria. So with that being said, why don't we table the motion on this one, and we'll bring it back for October for a final review and approval.
This is Virginia. Thanks, Donna. That sounds like a great plan.
This is Nancy. I second that motion to revisit and re-review in October.
Do you want me to capture that they approve this, bringing it back?
I don't think you have to, but you can if you want to.
All right, we'll leave it and make the motion in October.
Okay, shall we move on to the final topic?
This is Alex Park. I'm sorry to be a pest here, but if we if we are going to revisit the policy, I would appreciate if we would also ask the experts about the 2 years or less stipulation.
Absolutely, that was what I was thinking as well. We'll do that.
Thank you very much. Yeah, it's a drug that we don't really know how it works, and 130 something patients, not necessarily definitive. I totally understand we have to kind of go by the letter of the trials, and I thought the policy was well made to reflect that. But yeah, if we're going to talk to some experts, we should include that. Thank you.
Okay, so this is Virginia, and we'll move on to our final topic with Marissa. Ophthalmic agents, gene therapy, Luxturna.
Okay, hello. So the policy that I'll be presenting today is on Luxturna, which is a gene therapy. So just to give you kind of some background on the disease state. So retinitis pigmentosa is a type of inherited retinal dystrophy, which is characterized by progressive dysfunction and degeneration of the retina, which ultimately causes blindness. So Luxturna is, excuse me, sorry, my computer. So Luxturna is a gene therapy which is used to treat children and adults with an inherited form of vision loss that could result in blindness. Luxturna is the first directly administered gene therapy approved by the FDA that treats patients with confirmed biallelic RPE65 mutation associated with retinal dystrophy. What Luxturna does, the mechanism of action, is that it delivers a normal copy of the RPE65 protein to retinal cells, which augments reduced or absent levels of biologically active RPE65, which leads to normal production of the protein, which helps ultimately helps restore patients' vision loss. So if you could scroll down a little bit, Lita. So, right there. So this is the clinical criteria that was created for Luxturna, and it is based off of the phase 3 trial that was conducted for Luxturna. And so just to go through the criteria quickly, the patient is 1 to 65 years of age with a confirmed diagnosis of biallelic RPE65 mutations, and that would be confirmed via genetic testing. The patient has a visual acuity worse than 20/60 in both eyes, or visual field less than 20 degrees in any meridian. If you could scroll down. Perfect. The patient should have viable retinal cells, and verification must be documented. So in the phase 3 study, they did have, this was part of the inclusion criteria, and also number 2 that I just mentioned was part of the inclusion criteria as well. So that can be verified by an area of the retina within the posterior pole greater than 100 micron thickness, which can be shown on optical coherence tomography, or it can be a clinical exam which would show 3 or more disc areas of retina without atrophy, or the remaining visual field within 30 degrees of fixation as measured by that isopter. We are putting in criteria, or the criteria is that they had not previously received Luxturna gene therapy or RPE65 gene therapy, but Luxturna is the first therapy of its or product of its kind, so they can't have been previously treated with Luxturna. Patient has not had intraocular surgery within 6 months in the intended eyes, and it's recommended that it be prescribed and administered by an ophthalmologist or retinal surgeon who specializes in performing intraocular surgery. In the clinical trials, these are still ongoing clinical trials that are expected to be completed between 2024 and 2029. So for the reauthorization criteria, since it hasn't been studied, there is no reauthorization criteria. So if you could go down, Lita. We would only allow for this particular product 1 dose per eye per lifetime. And this is the HCPCS codes and the dosing for Luxturna for your reference. And then if you could scroll down, Lita. And then here's the authorization form that we would be asking providers for information. And if anyone has any questions, I'd be happy to answer them.
This is Virginia. Thanks, Marissa. Any questions from the committee? I don't want to move too fast. Okay. It doesn't look as if we have any stakeholders, so if there are and I'm missing you, make sure you raise your hand. And otherwise, there are no questions and no stakeholders. We'll move to the final motion.
So
This is Martha. Can you add a -1? Sorry. Thank you.
It's Alex Park here. Just looking at the FDA reference for this, Marissa, it looks like there's some sort of a steroid requirement that you have to be on that at the same time you take this. Did you see that in your run through?
I did. Yeah, I did have that. And initially when I was drafting the criteria, I did have that included. But I did get feedback that maybe it should be taken out. So are you recommending to add that back in?
Well, I'm just thinking we should be consistent. We've been fairly proscriptive in some of the other criteria about, phosphate or vitamin D, this or that being stopped 1 week before, et cetera. But I'm curious to know what you were thinking, what the thought process was behind taking it out, since it already seems like you thought about that requirement.
Yeah, this is Nancy. I have the same question. I guess what was the reasoning for, because you had it in before, and then I don't know what the reasoning is, or the thought process was for taking it out.
Hi, this is Marissa. I think I just took it out just because it was already on the FDA labeling. And I got recommendations to take it out, so it might have been redundant, but I can definitely add it back in.
So it's Alex Park. I think that'd be fine, just to be consistent with how we've been on some of the other policies that we've reviewed today. Thank you, Marissa.
No problem. Thank you for the recommendation. I will definitely add that back in.
This is Alex Park. I move that the Apple Health Medicaid program implement the clinical criteria listed on policy 86.37.00.-1 as recommended, adding back simultaneous steroid use.
This is Connie Nguyen. I second.
This is Virginia Bucola. All those in favor?
Aye.
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 Any opposed? Okay, the motion carries. And with that, we are ready to adjourn the Drug Utilization Review Board for today. Thanks to everybody for helping me keep on track. Thanks to Lita and the HPA for helping us do this virtually. So we'll look forward to seeing everybody again in August. And maybe we'll turn it over to Lita if there are any last minute comments.
This is Lita. Ginny, thank you. You did a great job.
You're welcome. Thanks. Once we figured out how to make sure I was doing the motions correctly.
Thank you, Jenny.
You're welcome, guys. It's good to see you all a little bit.
All right.
Thank you.
Bye, everyone. Stay safe.
Bye.
Bye.
Bye.